Bonnie, don't distract the students, please. Ready? We're going to start, I guess. We shouldn't wait. Any questions, first of all, what we did yesterday? So today is going to be a variant calling, but before we start about talking sequencing again and variant calling analysis, I wanted to review a couple of things we did yesterday because we didn't have time to go through all the exercises, right? We only did one notebook, but actually, if you look in the Jupyter notebook, there's another folder with two data sets, two samples. So we only processed one yesterday, but you have another folder with two samples, and the idea was to process both of them and analyze them, or do differentials in expression, comparing clusters between them. And I'm noticing my voice is getting worse and worse every day. What is it? Yes. So in the new sample, or the new folder, or the two samples, if you do a normal analysis, you would encounter this situation, right? Both of them are PB and Cs. So we would see the same type of clusters we saw yesterday, B cells, MK cells, and all that. But they don't overlap, right? So then is when we need to do integration. So this is the part we didn't see yesterday. The notebook is there. What I'm going to do is, all notebooks in general, I'll send them to you or figure out a way to share them with you. So if you have time, you can do that exercise. But you're going to need both notebooks because the second one, you don't have the same information because the idea was I'll let you do the analysis. But the first section of the second notebook should start the analysis for you, but then you need to finish it on your own. So any questions? No? Questions about the notebooks or in general? Okay. So today, variant calling. And today we have a special guest directly from Korea. Jamin Choi is going to give us a presentation. Jamin got his PhD from Maryland University. Probably about some boring topic, so it doesn't matter that part. He joined Yale in 2013. And then that's when the fun started, right? So he started doing NGS analysis, variant calling, in rare diseases and also in cancer. He did a lot of work in cancer too. He joined Rick Lifton Lab is when I met him. He stayed there for 5 years, and then he decided to go back to his homeland. He joined the Korea University as a professor. He's been working. He still works in. You still work in cancer, right? You also do cancer. He's not going to talk about cancer today. He's going to talk about rare diseases, a big cohort they have in Korea. So, ready? I think we can go. So, big welcome for Jamin Choi, our guest star today. Yeah. If you need power, that's the power. Yes, we're going to have the issue of changing the resolution. Yeah, I noticed. Don't worry about the color. We'll fix the color in a second. Oh, don't even start that. So stop it for a second. Yeah. Oh, hold on. Now? Mm-hmm. No.
Oh, nice.
We learn it through error. So you're gonna have to wear mine.
So I need to press.
Oh no, you have sorry. This you need to point it here, and you have a pointer also laser pointer.
Hello, everyone. Hi, everyone. My name is Jungmin Choi. Thanks for the kind introduction.
You can tell them what you did as a PhD.
Yeah, I did some boring thesis work on my PhD. So when I first started, by training, I'm a chemist. So I came to I came to America thinking that I would synthesize like whole different types of small molecules and then eventually conquer the other cancer with those new molecules. But then unfortunately, my first PhD advisor didn't get tenured. So I actually had to find a new advisor. Across the other hallway, I found the new young faculty who just published 3 science papers on parasite genome sequencing. So I thought, bioinformatics, that sounds pretty cool. So I joined his lab. That's where I started learning about all this next generation sequencing and stuff. So basically what I did was I spent my PhD sequencing the parasite genomes, so-called trypanosomes, Trypanosoma brucei, T. cruzi, Leishmania major. So these are the genome sequencing was done by my PhD advisor. And then after finishing my PhD, I joined Yale University, where I started working on human diseases, including rare diseases. And then even after I got my permanent job in Korea, I still felt my passion is on rare diseases. And I was lucky enough to involved in Korean Rare Disease Consortium. So today I'm going to, so it's been roughly around like 5 to 6 years since I got my permanent job in Korea. So I'm basically going to show you what I actually have done over the past 5 years today. Okay. I think everybody knows why we are interested in human DNA, right? So essentially what we do is looking at this. Okay, so basically what we typically do is we consider genomes as a blueprint of human health. And then we should all agree that human health, we need to understand human health and then human disease in order to conquer the other human diseases. Precision medicine, everybody is interested in is essentially reading the other human genome. And then in a way, it's a process of interpreting what's wrong with this human DNA. So it's been quite some time. I think more than 30 years ago, starting 1990, many scientists in the States realized that we need to know human DNA in order to understand the human health and disease. So they decided to basically sequence human DNA starting 1990, and then in 2003. Eventually it took more than 10 years, 13 years to get the first draft of human DNA, which is shown over here in 2003. But then anything like non-black color basically indicates that they are not fully resolved. So it took another 20 years from 2003 to 2022 to fully understand the human genome of 3 billion letters of them.
That was the telomere to telomere?
Telomere to telomere was done in 2022. So we thought, 2003, everybody was so excited that we have finally our human genome draft. We will be able to conquer all the other diseases. No, it took another 20 years to fully understand what's in there from telomere to telomere. how was it possible? Because of the other advances in sequencing technologies. So I told you that to complete the other human genome draft, it took 13 years. And then how much did it take to sequence it? $3 billion to sequence one human genome. So it was super expensive. And then it got pretty cheap right after next generation sequencing technologies emerged in the market in 2006. And then ever since 2015, the market was basically dominated by Illumina sequencing. And then whenever they made some advances in technologies, it got cheaper and cheaper. And in 2015, finally, human genome sequencing cost was pretty much plateaued around 1,000. And then since 2022, in the States, you can just pay $200 per human genome right now. So we are basically living in the other era where we can pay $200 to sequence human genome. And then what's going to happen after that? I think it's going to be even cheaper, but I don't think it's going to be like free. You need money to sequence something, right? But then I think it's going to be practically free for everybody because, for instance, like in the States, I know many big farmers are paying for your whole genome sequencing cost because your data is pretty important. In Korea, government is actually paying for this sequencing. They are actually giving us money. So if you volunteer to donate your blood, then Korean government is giving you $50 for that. So I think it's going to be eventually free. And then what's going to happen? Like, because it's free or it's getting so cheap, now everybody is starting to build their own nationwide biobank with that. Starting with the United States, I'm pretty sure you are all heard of UK Biobank study. So they initially, I think it's been more than 10 years, they started collecting the whole genome sequencing data from 500,000 individuals, right? And then United States, they started their own projects, which is called All of Us. China, Japan, they all started collecting their whole genome sequencing data. And Korean government also started doing pretty much the same thing, which we call National Project of Bio Big Data.
So it's only genomics or it's like classical genomics?
That's a good question. So this is the first big scale, large scale study launched by Korean government, and then we are not quite sure how many samples we can handle, how many samples we can produce. Do you have enough capacity to store it? Do you have enough capacity to analyze it? So as a pilot study, what we started was that we decided to sequence 25,000 whole genome sequencing first, and then decided to scale it up based on the other pilot study. So these are the number of individuals. I just told you that we decided to sequence 25,000 individuals as a pilot study. And luckily, out of 25,000 Korean individuals, 15,000 individuals were a rare disease cohort. So we decided to focus on rare disease cohorts as a pilot study and then see what we can handle. And then, based on this analysis results, the plan was to decide, are we going to scale it up or keep it as it is? So just going to briefly introduce how they are represented. So out of 15,000 individuals, female and male were, as a design, we wanted them to be equally represented. And then the cool thing about this study was that majority of them were recruited as trio. so that we can call de novo mutations. De novo mutations, for those who are not familiar with de novo mutations, these are the other mutations that are not present in healthy parents, but only present in the PRON. And many of the developmental diseases, we know that 30% of them are mostly attributed by de novo mutations. So these are the other mutations that are really important. And then so far in Korea, people didn't kind of realize, like de novo mutation is important. So they tend to only analyze probands only. But then with this pilot study, some of the other human geneticists were involved, and then we insisted that it has to be trios. So if we can, we analyze the other rare disease cohort as trios. If not, duos or singletons. And as many of you already know, rare disease, it is rare. But then the types of rare diseases are it could vary. So in our cases, majority of them were neurodevelopmental disorder patients, roughly around 3,000, followed by some tumor syndromic patients, cardiovascular disorders, and so on.
So in this, like immunological and hematological have been classified separately.
They were analyzed separately.
Because Korea has got a KeyNet is a Korean immunodeficiency network. In that only there are 200 patients.
Right. So that's the course. I'm not quite sure if they are overlapping over here or not. But then, as a pilot study, we actually didn't have people involved.
This data is from which year?
So this is from 2019 to 2022. And then we carefully selected the largest hospitals in Korea, 5 largest hospitals in Korea, including Samsung, Seoul National University, and then Severance, and so on. And then
KeyNet is a network of primary immunodeficiency disease, and that itself has got more than like around 150 to 200 patients.
Right. So the thing is that I think that's going to be very precious resources.
Are they represented here?
That I do not know. Yeah, I'm not quite sure. I'm not quite familiar with that consortium. But then I'm not actually talking this here, but then it was pretty complicated because of the other patient consent stuff. So many of the other Korean people, surprisingly or not, we are quite conservative. So they do not tend to reveal their genomic information. So surprisingly, even though like as a parent, even though my child is sick, they are happy to sequence their child DNA sequence, but then they are reluctant to sequence their own DNA. So I think it's kind of cultural reason. So it was quite difficult. There were like multiple layers of difficulties in sequencing data. So, as a person who was quite interested in rare disease cohort, and then it was a great opportunity because Frances just mentioned that in Korea, nobody is interested. I shouldn't say nobody, but then there are very few people interested in rare disease patients. So in 2019, when I first started my own lab, somebody at the other government high official came to my lab and then asked me, okay, what are you going to do in Korea? And then I basically told him, I was trained to analyze rare disease genomics, and I would like to do this in Korea. And then he was very surprised and then asked me, why should we spend taxpayers' precious money on rare diseases? There are so many cancer patients and then Alzheimer's disease patients and all this kind of stuff. So I felt pretty bad. I felt, there's no way that I could do really good rare disease genomics analysis in Korea because everybody's only thinking of cancer.
Even in India, the situation was the same.
Is that right?
But then there was a national rare disease policy because patients with rare diseases went to court and they said that it's their right to get the best treatment available. And then the government had to formulate a rare disease policy. And now there are actually rare disease centers in the country.
Right.
And I think the paper is in review, but I think for a large country like India, the database now has around 20,000 patients. The National Rare Disease Database in India has got 19,000 like your data, most of them are neurodevelopmental.
Right. So I was kind of doomed, and then, you know, I kind of knew that everybody's interested in cancer, genomics, for instance. There are only grants, you know, government grants about cancer, majority of them.
Cancers themselves can be caused by germ-like variants.
Yeah, that is true. So in a way, still I had to do, you know, work on the other cancer patients. But then suddenly I kind of realized that, you know, Korean government has a plan to sequence a rare disease cohort for whole genome sequencing data, which was good for me. So I was lucky enough to work on this 15,000 individuals. And what we got was that, you know, we realized that whole genome sequencing quality was pretty good. For instance, we are aiming for 30x coverage. They are all well above 30x. And then, you know, since they are Korean by definition, basically wanted to see if they are really Korean or not. And then based on this PCA analysis, they are largely overlapping with East Asian, not necessarily Korean, but then most likely they are, at least we know that they are East Asian. So they were produced pretty well. We have, you know, several pretty good sequencing companies in Korea. They were, you know, like voluntarily sequence them in a pretty short period. And then how did they store the other data? In Korea, we have a supercomputing center which is called KIST. All the other sequencing data were stored over here. And then because of the other privacy issues, they are, you know, they are housed within the other secured network. So for you, for anybody, even you can actually analyze the data if you want to. But then you're supposed to write a proposal to Korean NIH. And within this Korean NIH, there's a committee called KODA. They're going to review your proposal. And then once they approve your proposal, you can use VPN to have access to this KIST.
So there is similar, like in India also we've got like 10,000 genomes and it's called a Genome India database.
Right.
So even the Department of Biotechnology in India has started an initiative where you write a proposal according to your area of interest, and then you can query that Genome India database of 10,000 healthy Indian individuals.
Right.
It was done at centers across the country because India is a very genetically
Right.
homogeneous population. So it was done at 10 different centers across the country, and the data is there. But like as you said, you can access the data, but you have to write a proposal for that. Like what are your objectives
Right.
to query the Genome India database, similar to what you were
Right. So the thing is that I think of this is, you know, pretty cool having like 15,000 whole genome sequencing, you know, high quality, sitting somewhere in the other supercomputer center. As long as you have a great proposal, you can have access. But then the thing that I realized was that this wasn't really ideal because, you know, the power of this population genetics comes from, you know, when you compare your data to other data, for instance, like Indian Genome Project or UK Biobank, all of us, you know, sitting over here, and then you can actually analyze the data only within this local on-premise servers. You won't be able to compare like your data to other population genetics data. So, but then, you know, this was that because, you know, the patient consent issues, you won't be able to get this data out of here and then put it on the other cloud computing system. So what we realized was that, you know, we can, you know, for 25,000 individual whole genome sequencing data scale, we can produce it okay, I mean, pretty fast. We have somewhere to secure it. And then somehow we were able to analyze it even though it was kind of restricted environment. So everybody was happy. It was done within 3 years. And then starting 2024, last year, we scaled it up, and then we decided to sequence 700 or almost like 800,000 whole genome sequencing data additionally on top of that. And then eventually, you know, Korean government official likes number, so 1 million, they are aiming 1 million. It's
What is the population of South Korea?
it's 40, roughly around 50 million right now.
Quite a 1 million, 1
Yeah,
million will be a large population.
a large population for sure.
It's like 1 to
Yeah, exactly. And then out of, I'm not interested in these numbers. I'm not quite sure if we have enough money to sequence 1 million. But then out of those, roughly around 50 million will be rare disease cohort.
And how do they check that they are healthy? I mean, like there are criteria?
That's another big topic. I'm not following it. There are huge discussions and debates among clinicians. How do we set the other, like objective criteria for being, because,
So-called
exactly, yeah, because there are no normal individuals, right? In a way, everybody's looking healthy, but then, how do we know they are normal, right? But then, I'm not getting into this, but so we already have sequenced it because it started last year. We have sequenced quite some of them. And then for the other pilot study, like Amit asked, it was all whole genome sequencing data. And then one lesson from pilot study was that whole genome sequencing data is cool. It gives us a lot. But then without having transcriptomics, proteomics, metabolomics, all other multi-omics data, it's still very difficult to pinpoint the cause of the disease. So we learned it from the other pilot study. For the main study, main projects, we decided to add these multi-omics data, even though the numbers are fairly small. But then I think it was a big leap for us to realize that whole genome sequencing alone is not the answer.
You finish one problem, you are ready for the next one.
So these are like roughly planned. So up until 2028, we are going to collect roughly around like 300,000 individuals whole genome sequencing data.
One center, right?
No, it's not. It's the other national project. So it's going to involve multiple hospitals.
The pilot was done at one center?
No, it was involving 5 centers. So it's great. I'm very happy that soon we already have 15,000 whole genome sequencing data for rare disease cohort. And then soon enough, we are going to collect 500,000 whole genome sequencing data from rare disease cohort, Korean rare disease cohort. On top of that, we are also going to generate some multi-omics data. And then with that, what are we going to do? What I do, and then what Francesc is doing, is essentially finding typos from our DNA. So if you were to go through this typos, so we know that there are 3 billion letters in our DNA. And then we are basically looking for misspelled letters, for instance, something like this. And then I know that we are doing this pretty well. So assuming that we already know the answer, there are several options to look for misspelled letters. For instance, like algorithm which is called GIAN can do this very well, like 99.9%. We can find it very accurately and then sensitively. Indel's a little bit lower, but then still we are doing this pretty good. So once we have one individual's whole genome sequencing data, we can find the mutations, misspelled letter pretty well. Then what's the answer? What's the issue? I think I just told you that I was very excited to have like a 1 million whole genome sequencing data, but then too many data is causing the issue. So I wanted to just point out several issues over here. First of all, storage. So I'm pretty sure you are all familiar with the whole genome sequencing data would produce roughly around 150 gigabytes of data. And then imagine that we are going to have 1 million whole genome sequencing data. The size of the storage has to be enormous, right? Computational power. I just mentioned that the PD of pilot's Korean rare disease course study was that, since it was housed within the other restricted server, there's no way to compare it to other cohorts, right? So having that said, computational power is very important. Even though we can actually aggregate multiple different cohorts from different projects, you should have very good computational power to merge them and then analyze them all together. And then collaboration. So back in those days, when I was actually working at YCGA with Francesc, the way we collaborate was that, say, like, Francesc has some cohort, and then I have another cohort, and we want to merge, integrate them, and then work together. What we typically did was we store the other data into the other external hard drive, for instance, or USB drive, and then just toss it to the other. In a way, we can actually merge them together and then analyze them together. Imagine that we are talking about one whole genome sequencing data of 150 gigabytes of data, right? Say we have 10 individuals. We are talking about a few terabytes of data. There's no way that we can actually put this in the other hard drive. Even though we are using, like Dropbox or Google Drive, it's going to take forever, right? 10 whole genome sequencing data. We are talking about like 1 million, few 100,000 individuals. There's no way. So no longer we can transfer the data. It has to be, the data has to be centrally housed somewhere, and then individual investigator should have access to that same data, right? So collaboration, accessibility, it has to be, your data has to sit somewhere centrally, and then as long as we have all internet access, we should go find them. That's the easiest and then most logical way to.
Is there any move towards that kind of a global consortium?
Yeah, I think everybody's doing this. And then this is why everybody's now, the only solution is cloud computing. So we should, there are some issues, for instance, like privacy issues and patients' consents and all this kind of stuff. Once this is all cleared out, I think the only solution is to put your data on the other cloud and then have approved individual users access to that data. And then data security, like, in Korea, this is one of the other biggest issues because every hospital has their own servers somewhere, and then they are always breached out by the other hackers. In Korea, like, there are quite a lot of hackers who breached out the individuals' whole genome sequencing data and then sell it to the other health insurance company in Korea. So what I'm suggesting to resolve these issues, I think the only way is to use cloud computing service. So everybody, I'm pretty sure you are all familiar with the other Amazon Web Services, Microsoft Azure. There are multiple. Google has their own cloud computing service system. In Korea, we also have a couple of Korean cloud computing systems, including Kakao Cloud, which I'm working on. So I think this is the only way. Like, you put your data centrally. There are some pros and cons. So this is the only way to handle like hundreds of thousands whole genome sequencing data. There are no other way to get the other enough computational power. Storage-wise, it's basically the same. And then this is one way to make our collaboration very efficient and productive. We know this because it became pretty successful based on the other experience from Anvil projects and then UK Biobank. They are all, Anvil is using Google Cloud Computing System. UK Biobank is using AWS currently. So in Korea, there are quite a lot of people pushing the other government that we should all put this rare disease cohort on the other cloud. But then these are some, there are some political issues. For instance, I just told you that there are Korean supercomputing center, and then the employees of Korean supercomputing centers are not happy with the usage of cloud computing, because in a way they are losing their like power to govern the other data. So they are like really against it, and then there are some clinicians concerned about the patient privacy issues. So with that, I'm going to briefly introduce what we actually have done with the pilot study data. And I told you that my passion lies on rare disease genomics. I'm not quite sure if you actually do use the same thing, but then in Korea and then in America, this is typical saying in the medical. Is that right? Yeah. So when you hear hoofbeats, you should think of horse, right? Not the other zebra. But then, as a person who works on a rare disease cohort, I claim that sometimes still it could be a zebra, right? So you should pay more attention to the other rare disease patients. So these are like, first slide that I always use to convince government officials that we should spend taxpayers' money on rare disease patients, because it's rare, right? I mean, as a definition in Korea, rare disease is considered as when it affects fewer than 1 out of 2,000, rare. But then there are so many rare diseases out there. Roughly around 5% of the whole population are affected by rare disease. And then as a geneticist, since majority of this rare disease can be explained by genetic cause, 72%, and then 70% of the other genetic rare disease starts in childhood. Once they have really early onset, many of them doesn't go below above 5 years. And then I think this is probably the most empathetic things that made me interested in rare diseases, is that like you get like ill, you do not know why you're not feeling well. You go to the local hospital, and then since this is a rare disease, they do not know by training. So they would suggest you to, you should probably go to a bigger city. Then they would go to the other bigger city. They are represented, and then apparently your doctor would say, I actually haven't seen it. I'm not quite sure. You should probably go to Delhi, right? And then they would send the patient to Delhi, and then in the other, university hospital, they would say, I'm not quite sure. You should probably go to see specialized rare disease center, right? So typical years to get diagnosed. I'm not talking about the other years to get treated. To know why you're sick, it takes around like 5 years. So it's a huge pain for the other patient itself, but then for the other patient family as well, right? It's a huge socio-economic burden for them. So I think we should pay attention to this rare disease patients. And then sadly, because...
Is it all cancers combined? Like, this statement is quite bold.
What is it?
Like
Yeah. This, yeah. So it's surprisingly more than that. But then I took it from the peer-reviewed paper. It's more than combined cancer and then AIDS patients. All cancers, yeah. So there are no available treatment options currently.
For some there are, so it is also important to know.
Yeah. So many of these patients are undiagnosed. So I think if you so this is something that I would say, like why do we need to do rare disease genomics, right? In a way, so far the current diagnostics were done basically like based on the the disease presentation or phenotypes. But then to diagnose the other rare disease more consistently, it has to be based on the other molecular level. And then to understand the other molecular mechanisms very precisely, that's the first step to get into the other discovery of targets.
Is this definition like varying from country to country? 2000 seems to be very low. 1 in 2000?
It is variable from
country to country. That's based on the other Korean definition.
WHO has got a different definition.
Okay.
So how you define a rare disease is also not consistent.
Right.
All right. So I joined Yale University for my postdoc training, and then this is essentially where I was exposed to rare disease genomics. Before then, like I told you, I was sequencing parasite genomes, and then at Yale, Bonnie is here, Frances is here. Yale Center for YCGA, genome analysis, was one of the NIH-supported Center for Mendelian Genomics. Am I correct? Yeah. So I still remember, like we were given many different types of Mendelian genomics sequencing data, and then asked to find the causal variants. And then every time we find something, we report it. If we are lucky enough, then we could get published. And then at the end of every year, I remember Francis was always emailing me, asking me to write down the list of publications so that they can report it back to NIH. Am I correct? So we did it. And every year, I was very kind of like bothered by these emails asking for the reports. But then, in a way, I think over the past 10 years, with the other efforts of many different people, we sequenced more than 75,000 whole exome sequencing data here. And then with this, we were able to find roughly around 4,000 genes that are associated with the other Mendelian diseases, indicating that if you have mutations, if you have deleterious pathogenic mutations in this gene, then you can now predict you're going to have onset of this disease or phenotypes, right? So with this Mendelian Genomics Consortium, out of 20,000 genes where human is supposed to have, we roughly now know 4,000 genotype and phenotype correlation. Then people would naturally ask, what's going to happen to the others? Are we happy with 4,000? Apparently not. Clinicians want more, right? For instance, like if you were to take a look at the other diagnostic yields, typically it's around like 30%. I mean, it varies depending on the other disorders. But then, for example, neurodevelopmental disorders, say like if you have whole exome sequencing data, the odds of having this rare disease patient molecularly diagnosed is roughly around like 40%. And 60%, the rest of them, you still do not know why this individual is sick, even though whole exome sequencing is available. And then we were thinking about this. Why can we diagnose patients precisely? And then we could think of some reasons. For instance, like with whole exome sequencing data, we are basically looking only at 2 to 3% of the other whole genome, which are protein coding. So, there's plausibly, it could be like non-coding variants, some, variants within the other deep intronic regions, or some tandem repeats could be causal for this rare diseases. So these are all type of mutations that whole exome sequencing cannot detect precisely. So we thought of, we should probably do whole genome sequencing. And by the time, the timing was right. Previously, whole genome sequencing was very expensive. And now the pricing for whole genome sequencing is getting decreased dramatically. So in my case, I have, like I mentioned, we have 3,000 whole genome sequencing data available from Korean rare disease cohort pilot study. We decided to look into the non-coding variants. And we started with the small nuclear RNA. We basically, we are looking at different classes of non-coding variants, but then we decided to look into small nuclear RNA first, and then what we actually have done was that we collected undiagnosed neurodevelopmental disorder patients, which is shown basically over here. So we collected Korean government study, and then from the private hospital, Seoul National University Hospital, we were also able to get additional 5,000 whole genome sequencing data. So as a total, we were able to collect roughly around 20,000 whole genome sequencing data from undiagnosed neurodevelopmental disorder patients. And then as a control, by then we were able to collect roughly around like 7,000 healthy Korean individual whole genome sequencing data. And then out of those, if you were to remove all the other healthy individuals, healthy parents, and then related individuals, we were able to compile.
Why is it called Jeju? It is from that Jeju island?
Yeah, Jeju. So they collected how many? 10,000 individuals from Jeju Island, and out of those, 3,000 was healthy individuals. So this was, so the issue of our Korean project is that like each city has their own projects. So Jeju, for instance, this is a really nice vacation plot where Francesc and I also went there for a conference. Yeah. So they decided to sequence 10,000 individuals of their own people. And then Korea 4K, even though they say Korea 4K, it was also another city in Korea. They sequenced 4,000 individuals there. And then it was roughly around like 7,000. And then we decided to remove all the other low quality whole genome sequencing data. After that, it removed roughly around 200 individuals. So as I mentioned, I focused on small nuclear RNA. And then among those, what we found interesting was that we found some interesting variants from RNU4-2. RNU4-2 is one of the other main component of spliceosomes. The machines that are involved in splicing events. And then people already have investigated on this RNU4-2 that there are some 18 base pair variant region, which is very important for keeping the stability of the spliceosome complex. And then this is where we found interesting variants. So now if you were to look at this plot, on top, this is, so first of all, what we did was that we called de novo mutations. Majority of our the cohorts were recruited as trio. So we were able to call de novo mutations. So what we did was that for every small nuclear RNA, we try to map de novo mutations from patients and then healthy individuals. So on top, you're going to see like large bars indicate there are pile-ups of de novo mutations in these regions. So they are here, here, here. And then in the bottom, basically de novo mutations from the other healthy individuals. It's basically like the other mirror image, indicating that this de novo mutation probably is not pathogenic because it is found from a healthy individual. Same here and here. But then within this region, critical 18 base pair region, we found that these piles of de novo mutations were only found from patients, indicating this is very likely pathogenic. Surprisingly, what we found was that there were exactly the same recurrent mutations found from 18 individuals having exactly the same de novo mutations. And then there were some 3 other mutations. So we found 20 de novo mutations from undiagnosed neurodevelopmental disorder patients having pretty much clustered mutation events in here.
Are these like symptomatic or?
No, so these were all, so I'm going to get into this symptoms later. So, so this is great. I mean, basically it can explain roughly around 0.72%. So if you were to just consider only one recurrent mutation, having it explain 0.61% of the whole Korean undiagnosed neurodevelopmental disease is a huge thing. Because we were like analyzing them as like a crude undiagnosed neurodevelopmental disorder patients, we basically wanted to see, what kind of phenotypes we could observe from these patients. They were mostly microcephaly patients. Apparently, they are having this microcephaly phenotypes. They were also having intellectual disability disorder. So we kind of know, even though we do not know the exact mechanism, we kind of know that having this recurrent mutation, recurrent de novo mutation, which was insertion of a single A to this position, would make you have like microcephaly. Another thing that we found quite interesting about this de novo mutation, having like de novo mutations in neurodevelopment disorder is not something like novel. Everybody knew, like you probably have heard of like autism example, right? As father ages every year, like having your child de novo, autism kids, the probability is getting higher and higher because of the de novo mutations originating from the father. But then, so like we all know that typical developmental disorder patients, 30% of the patients are owing to the paternal de novo mutations. It's very well known. One thing that we found quite interesting was that we thought of the same thing, like de novo mutations, microcephaly must be father, right? But then, very interestingly, all the other 100% of 20 de novo mutation, they are all from the mother, which is very interesting, and we do not know what it means. We are speculating that somehow this de novo mutation is causing some, strong positive selections in the oocyte maturation development. We do not know what is actually causing this, but then this is something that was unexpected. Another thing that we are quite interested in was that, having this recurrent mutation, it was all insertion of a single A, right? And then this is causing the instability of spliceosome complex. But then another thing that you can think of is that, why should only insertion of A, right? There's a strong selection of A. Could it be insertion of T or insertion of C, which could cause the instability of spliceosome complex? We couldn't understand. So we basically like simulated like different scenarios, and then took a look at the secondary structures, and then some stability simulations and stuff. To be honest, we still do not know why exactly causing this. But then something interesting that we found was that compared to the wild type, only when we inserted a single A mutations within in this locus, it was actually causing the increase of entropy, indicating they are more unstable. But then, for somehow, if you were to add another nucleotide, or C or T, it was basically like a wild type. So still do not know what's behind this, but then I think we think this is definitely affecting the other stability of the other complex, which is causing the other issues in proper splicing events. So to test this hypothesis, what we did was we collected the blood from patients and then their healthy parents. And then we did RNA sequencing to see what kind of differences we would get from patients versus healthy parents. And then these are the other summary of the other results. Something that has been expected was chromatin remodeling pathways, because it was well, chromatin remodeler is a well-known cause for the other neurodevelopmental disorders. So we are somehow expecting this, like if you have like, alternative splicing events from chromatin modifiers, then probably you would get microcephaly phenotypes. But then something that we are not expecting from this analysis was immune response. So in the other microcephaly patients, there are higher expressions of, if you can see, IgH, many different genes indicating activated immune response. In a way, this is plausible because, say, like you have some unexpected splicing events happening in your system, then you would get your immune system activated. But then we are quite not sure if this is a primary results from the splicing on alternative splicing events, or the secondary events from these other pathways. So this has to be investigated further. And lastly, we took so far I told you that this insertion of A is causing instability in this spliceosome complex. So naturally, we would think that this should actually result in alternative splicing events. So we took a look at it. Indeed, especially within the other 5' splicing events were alternately resulted. And then again, this was most frequently seen in the genes involved in immune response, DNA repair, and then chromatin biology. So what's going on? So we basically pinpointed something quite interesting from undiagnosed neurodevelopmental disorder patients that were previously unappreciated, like non-coding variants. But then, like, we found it, but then we still do not know the other biology behind it. Why this is causing microcephaly, intellectual disorders. So in order to fully understand the other mechanisms, we are doing some collaborations using brain organoids and then mouse models to fully elucidate the mechanisms. Another thing that which went well was that because these patients were analyzed from the other cloud computing system, so we basically presented this data in the other conference, and then Dr. Banka from Manchester University approached us saying they found something similar from other genes, RNU2-2P and then RNU5B-1, which is also one of the other major components in spliceosome complex. And then they basically told us that they found like recurrent de novo mutations in these genes. Can you take a look at these de novo mutations from Korean populations? And as expected, we found even though we didn't find a lot of patients because of the other core size was much smaller than Genomic England project, but then we found it, and then we were able to put this together and then we got published. So I think this is another example where we need to utilize a cloud computing system, because otherwise collaboration within this scale of a large genomics cohort, it's impossible. And then for the next 10 minutes or so, I'm going to introduce, like, using whole genome sequencing data is pretty good, promising. You can help diagnose many undiagnosed rare disease patients. But then there are some other categories that are getting popularity. So whole genome sequencing data so far that I presented to you is all based on Illumina sequencing. Typically, the size of sequencing reads is around like 150 base pair. 150 base pair, we consider this as pretty short. So in other words, we call Illumina sequencing as short read sequencing. I think on the other pipeline to analyze this short read sequencing data, whole exome sequencing or whole genome sequencing, I think we are doing this pretty well. In one paper from 2022, they demonstrated that from taking the other blood out of the other patients to analyze the data, it took only around like 13 hours. You can basically sequence it and then analyze it within a day, which is incredible. But then, like I told you, our diagnostic rate is still somewhat disappointing. It's around like 40%, even though we are using whole genome sequencing data. Non-coding variants, I don't think this is actually solving all the other issues. So then what? What are we missing? And then people thought of it initially, it has to be non-coding variants, but then we know that this is not non-coding variant because we cannot solve all the other cases. Now they are thinking that there must be some variants that cannot be resolved by short-read sequencing. And that could be some of the other repeat expansions. So I'm pretty sure you're quite familiar with the sequencing technologies provided by PacBio or Oxford nanopore sequencing, represented by long-read sequencing. Now, instead of sequencing 150 base pair, you can sequence hundreds of thousand base pairs at once, which apparently is going to help you to precisely identify very complex structural variants as well as many short tandem repeats. And then especially these long-read sequencing long-read sequencing technologies can also offer methylation status as well. So we thought of these are something that we are missing. So even though we can detect small indels or SMP SNPs pretty well using short-read sequencing, majority of other variants within our human genomes are structural variation, inversions, copy number changes, and methylations. We are thinking that this could be some revenue that we should look into. So far, short-read sequencing has pretty good advantages in assembly, small indel, copy number variation in some occasions, mitochondrial variants for whole genome sequencing, but then all other area, repeat expansions, mosaicism, complex structure variation, methylations can be obtained only from long-read sequencing. So what we did was that we decided to sequence some of the other undiagnosed rare disease patients that were not solved by either whole exome sequencing or whole genome sequencing by PacBio, long-read sequencing. And we especially focused on this short tandem repeats, first of all. Short tandem repeats by definition, repeats of typical 3 nucleotides. And then we focused on this NIID disease, which is which was found fairly recently. It was first described in 2019, initially described by Japanese colleagues, and then later on it was also reported in Chinese. So we think this is this disease is prevalent in East Asian population. These are very rare neurodegenerative disorders, and then known pathogenesis is caused by short tandem repeats in Notch2 NLC gene, 3' UTR. So what we did, knowing that we have 3,000 Korean rare disease patients that are unrelated, what we did was that we took a look at Notch2 NLC and then took a look at the other distributions of this short tandem repeats. And then we were able to find some of the other patients had outliers, indicating that they are long expanded short tandem repeats in Notch2 NLC regions. So we focus on these 6 putative individuals. And then what we found was that looking into these 6 individuals, so these are 6 individuals, everybody except for 1 individual having presenting metabolic disorders, they were all falling into the descriptions of NIID, the disease that we are quite interested in. So we thought that this must be the diagnosis for this undiagnosed rare disease patients. And luckily, all others, we didn't actually have a detailed information of these patients because of the other patient privacy issues. But then luckily, the very first patients having more than 100 repeats, this patient was the other incoming patients for Seoul National University Hospital, the hospital that we are closely collaborating. So we looked into this patient's clinical information. So these are the pedigree of these patients. So there's a healthy father, healthy mom, and then these are the other patients where we found more than 100 short tandem repeats. And then this proband actually had an elder sister who died out of this disease. And then before she died, we whole genome sequenced her, and then we identified roughly around 90 short tandem repeats here. So, like one thing that we found interesting was that so we had like short sequencing rate from whole genome sequencing data, and then we use expansion hunter to predict the other number of short tandem repeats, resulting in this. And then we are also using long read sequencing. In this case, we are using both PacBio and then Oxford Nanopore technologies. And what we found was that we thought, based on the whole genome sequencing data, we thought father actually had short number of short tandem repeats, but then long read sequencing indicates otherwise. Oxford Nanopore Technologies indicates that instead of having 57 short tandem repeats in healthy father, he actually had roughly around 6,000 short tandem repeats in here, which puzzled us. How come he is healthy, right? Having this many short tandem repeats, he cannot be healthy. So what we thought was that so first of all, we basically wanted to see if this is true, that father, even though he is healthy, he had roughly around like 6,000 short tandem repeats. We wanted to make sure that this repeat was transmitted to both children. And then after phasing the other mutations, indeed, it was actually from father, not from the other healthy mom. Then how can we explain this 6,000, having like 6,000 and then being healthy? How can it be explained? Luckily, what we were doing some literature searches, and then there were some other examples of neurodevelopmental disorder patients having large number of short tandem repeats, but then onset was somewhat predisposed because of the methylation status. So as I told you, like this long read sequencing technologies also provides you with the status of methylation. So what we found was that even, although father having this large expansion of STR, his methylation was, I mean, the promoters of this gene was heavily methylated, hypermethylated for the other father. And then for the other daughter, methylation was hypomethylated. And then we think that even though this sister only had like 90, which is actually shorter than the other proben, the onset was much faster, and then disease was much more severe. So and then proben, although he's displaying these symptoms, the other methylation was somewhat moderate. So I think still he is actually displaying the other onset, and then the symptom is somewhat mild, and he is still surviving. So I think this is something that we kind of figured that like whole genome sequencing itself is pretty precious, but then whole genome sequencing alone is not enough. We need to do like, in addition to short-read sequencing technologies, we should be able to utilize long-read sequencing as well as methylation. We are currently doing RNA sequencing just to make sure that this is methylation is the proper mechanisms that as we are suspecting. So I'm not quite sure how long it took, but then for the last 1 hour or so, I basically wanted to tell you that large scale whole genome sequencing analysis can indeed increase the diagnostic rates for many rare disease patients. And then so far, many countries have focused on generating only one whole genome sequencing data, which is, I think great. But then from the other pilot study of many different rare disease patients, we also get to realize that whole genome sequencing, we shouldn't rely on just one omics. We should produce like multi-omics data, especially transcriptomics data is essential. So you should always work on whole genome sequencing data as well as transcriptomics data. And then lastly, I wanted to emphasize that cloud computing system is probably not the only way, but then the most plausible way to do large-scale, genomics analysis. There are some disadvantages, which is the cost. It's very, very costly. I'm getting to realize it, but then I'm hoping that the cost would get decreased every year, and then hopefully we can do some great work with the collaboration between other nations and projects using cloud computing system. Okay, I think that's basically it. Thank you for your attention and happy to answer any questions.
Questions? Actually, Bonnie can give you more details, but even methylation in RNA can resolve phenotype, right?
Yeah.
So it's not like methylation in the DNA sequence. Now we need to look at methylation in the RNA sequence to figure out phenotypes. It's getting more complex and more complex.
Right. So that's a good point. So as far as I remember, we actually haven't produced the other data, but then Oxford Nanopore. So like we always say like we are sequencing RNA, but then I always tell my students we are not sequencing still RNA. We are sequencing cDNA. We are still sequencing cDNA. There are the only technologies that I know of that can directly sequence RNA is Oxford Nanopore technologies. You can directly sequence RNA, which is great because, like Francesc mentioned, we can also detect the other modifications on the other RNA, which can give us a lot of information.
And one other thing is like I was really intrigued to see like you guys are using methylation, DNA methylation, and that is the biggest challenge. In US, they haven't started still using DNA methylation for clinical, and in Yale I was trying variant in a gene. So DNA sequencing is only 30%. Remaining 70% of variation of the disease is manifested through methylation changes, and there are two locus which are known to drive those changes. And so if you are not looking at those methylation changes, you have a phenotype, but there is no way you will be able to find the causal reason for that. So I think that is why we are missing a lot by not using multi-omics as an approach.
Yeah, he pointed out that to
No, I'm saying like this is something that we always think that people are not using it. And that is last statement.
questions. Thank you very much.
Thank you.
So, probably we can have a tea and then tea break, and we can start after that. So we don't lose any time rather than just let it up.
Hi.
So you showed that because of the insertion of A in RNU4,
For this too.
so that's leading to the change in the expression profile in immune cells, blood immune cells. So that's
you for answering. So I guess you recognize that title, right? It was a me. So it's that person at the end of the room that has a problem.
There is an interviewer bias.
Inter
Like the person asking the question is oriented towards something already.
oh yeah, no, for sure. I'm clear in my question and in my answers which one is the correct out of the three. Oh, here we go. Oop, oop. Okay. So Jaming talked about something really interesting, which was really interesting for me, right? The idea of you have a single space where all the data sits and people connect to it and people can analyze it if you have the right permissions, right? And I don't know if that just started or not with that CMG project, the Center for Mendelian Genomics project. At the beginning, every center had their own data. It was complicated to share. And by the end of the project, they decided, okay, let's use the Anvil project. That was the idea of putting everything in one spot. And the only thing you need to do is connect to it and do analysis. The tools will be already there. So you don't have to install anything. Just connect, tools are available, and you do your analysis. But I think the main problem also of that system is that the payment system, you needed to pay. Somebody needs to pay that infrastructure. And it seems it's not that easy. They use one system called Terra, and you can kind of predict how much you're going to spend. But I don't know, how many times do you need to repeat an analysis? 100 sometimes, right? So it's almost impossible to predict how much money you're going to spend. And requesting soft money for this thing seems harder than requesting, oh, I'm going to spend some money in a computing center and do the analysis in my computing center. So it sounds good, but I don't know how easy it is to implement or how many people is actually using it. I don't know, in the case of Korea, I don't know how it's going to work, but the Anvil project, I'm not that convinced. Because the problem is when you create a new barrier, which is the payment system. We have to move all the data that we had in all centers to the Anvil. And from that moment on, I don't know, our centers never use it.
And you lose all your quality of the data then?
If we use
You lose the quality of the data if you put it on
the quality in the sense,
the quality in the sense that you have no holders that data.
yeah, I mean, we already, that's the thing. It was already public because we have, it was already submitted to NCBI, right, to the SRA, the short read archive. But then we needed to move everything to Anvil to make it centralized. And then we could add more data to that because then you could fine tune the consent, right? So if it was, let's say, only for researching on X, then you can really fine tune who has access to that data. I understand the idea is really good, but I think at the same time you create a new barrier with how you're going to pay for it, I guess. That was my whole point. So I had a few slides to overview. I mean, we have a really good example. You've seen, we've been talking through these days about variant calling, right? There's different talks that talk about it yesterday, today, Jiangming. So I'm going to deal more about the technical part. I probably have way too many slides here, so I'll try to go a little bit fast so we can go directly to hands-on. Do I need to tell you what is variant calling? You probably already know. So it's basically finding the difference with some genomic sequence that you have as a reference, right? You already know this process too. Unlike transcriptomics, now we care about the sequence. Transcriptomics, we only want to align it. Once you align it, you want the count data, right? That's what we know. Now we don't have any count data, but we have a sequence and we want to see the difference. So the alignment process is really important too, and how we do it. And then we have another process, which is the variant detection. So pretty much we could split it globally in those two steps, sequencing, alignment, and variant calling. We have two big types, or more than two big types, but there are different types of variants. So,
So the alignment in transcriptomic data would not be very strict because you just need to.
well, it is strict, right? But it's in the sense that,
For example, here you are trying to call as SNV, so it has to be very
yep.
stricter than, I mean.
And the length of the sequence is more important, definitely, right? So you want the longer the better, so you can cover as much as you can. With transcriptomics, you can work from probably 40 bases up to 150. Maybe for what you want, the result is going to be the same. In here, definitely, you need to pay more attention about sequencing, length, and precision of the alignment, as you say, right? You need.
Alignment algorithms are the same, whether you use.
Yeah. I mean, the alignment algorithms are pretty much the same, yeah, or at least are based on the same things, some of them. Oh, yeah, so I was trying to talk that, yeah, so we have different types. We can talk about single nucleotide variants, structural variants, or even copy number variation variants, and combination of those. And Jianming talked about all of them, I think, right? Structural variants in general, even copy number, we put them aside a little bit because you may need different tools for analysis. So we're going to focus on single nucleotide variants today, but anything that's also an artificial definition. But generally we talk everything above 50 nucleotides, so it's affecting 50 nucleotides, we classify them as a structural variants. And also you have different types. Again, today is going to be about single nucleotide variants, not about structural variants, but they exist and they have their specialized tools. Because it's a slightly different problem, right? And that's another interesting thing, which is we are not perfect, right? And surprisingly, we are quite imperfect. There's 3.1 billion bases. We have around 20,000 genes, as we're discussing these days. The ENCODE project, I don't know how old is this data, but they estimate that 40 to 60% of our genome encodes for something. Generally, on average, right, each person has around that 3.5 million SNVs, 500,000 of small indels. This is another interesting thing. On average, between us, we have around, which we never, or we kind of ignore a lot of times, but among us, so whatever we use as a reference, we assume is the reference, but there are a lot of large rearrangements between us and that reference, or among us. So between 1,500 and 2,500 on average of large rearrangements in our genomes. So when we talk about, generally we say, oh, it's 99.9 equal, the sequence. Yeah, the sequence it is, but we have a lot of large rearrangements that also create some differences. And this is always the part that interests me the most. So we are born already with around, let's say, 300 loss of function variants. Loss of function meaning that gene is coding for something that is wrong. So we already have them. So if we do an analysis, you're going to find variants that they are going to be classified as pathogenic. In general terms, I'm going to call it, but well, better general terms, they're going to be classified as loss of function. So we have them. It could be that it needs to be, that you can have only one copy and the other copy is completely normal, and that's enough. But even if we have a gene that is completely, we have a loss of function of a gene, of a complete gene, the organisms have ways to compensate for that. So, and there are a lot of systems that have been studied, I don't even remember, but all of them, but there are different ways that the body has to compensate. So what I'm trying to say, yeah, so if you try to do an analysis of let's say, a normal person, you're going to find that that person has already 300 loss of function mutations on average. And even what I was trying to explain before, that between even some of those, they're going to be associated to some disorder. So can you give me ideas? You don't have to answer through the computer because it's a bit painful. But can you give me ideas why we want to study variants? One of them is rare diseases. We know that, right? Any other? No, you don't have to go to the website. Precision medicine, yeah, pharmacogenomics, right? All related to how we can adapt, or be specific about the treatment.
Organ transplantation.
Organ transplantation, we saw an example, right?
Association
Yeah,
with
population genetics.
association with many diseases.
Yeah, I mean, it doesn't need to be rare diseases. Diseases in general, right? We can study them. It could be a common disease, which is a harder problem. We talk about autism, right?
Ankylosing spondylitis, we know the association
Yep.
with that genetic.
In
Will lead to some diseases in future, sir.
yeah, I mean,
Best case.
they are good answers. So I always, when I try to think about applications, it always happens the same thing. I put some and there must be others, and then is when I write a question for the audience, outsourcing to the audience, because probably you have better ideas. I think we pretty much mentioned all of them. Oh, cancer research, yeah, for sure, but it's related to any diagnosis, right? Population genetics, agriculture, ancient DNA studies, forensics. Forensics, yeah. So actually, okay, I'm going to play Bonnie's role now. I have a story to tell. So I did ancient DNA long ago. I work in ancient DNA, like 25 years ago or so. And in forensics too, there were a company, I think it was in Netherlands, it was a cosmetic company that was working in shaving brushes. And the high quality shaving brushes, they use badger hair. You know, a badger, it's, yeah. And then it's the species name, there are different ones, but it's Meles, the genus is Meles, something else. So they work with that, and they were worried because they were getting hair from China, or the fur from China, and China, some of species are protected. So they were really worried about that. So they sent us the brushes, and we did the study and everything. So the company ended up suing another company in China for sending protected products to the Netherlands. I think it was a year later, CSI, you know CSI, the TV show, the criminal that they do forensics, they did the same thing. So then I discovered the CSI read papers about things, and then they do episodes. So I like to think that they base the episode on our paper. Sorry, what? Oh, no. But I mean, so they changed it. It wasn't a badger. It was a painter, right? A painter that left the hair in somewhere, and it was hair from a badger, and the forensics did the analysis, and they discovered that. Yeah. I think, yeah, Bonnie also mentioned this paper. We are proud at the Genomic Center about this paper because it was the first application in whole exome sequencing in clinical settings, right? It wasn't the first paper in whole exome sequencing. I mean, there were 3 papers being worked at the same time. Jay Shenduri was the first one. This was the second one. And then Kaya, which was another investigator. Whole exome, it was Nimblegen, or the first Nimblegen platforms that were available. Or it was a collaboration, I guess, with Nimblegen. And Kaya, which was also at some point work at the Genome Center, was the third paper. Also in a rare disease. Of course, you're gonna need to do sampling in your projects, and based on your goal, you're gonna do different samplings. Especially, I wanted to mention the difference between, and I'm probably going to mention that later, but if you work with somatic variants, you generally sample the individual twice, the tumor and the normal match, because you're going to have to distinguish what is a germline, what is a somatic. For that, you need both samples. And there are different types of studies, right? You can work with trios, you can work with big cohorts, like Jianming's research he showed today. You can work with families or even populations, depending on your goals. Sequencing, we've been talking about a lot of this. Based on the cost, and Jianming mentioned, we can give him different prices. He gives 200, I'm giving 300. I don't know what Bonnie gives, but around those numbers, right? But whole exome sequencing, so this is not necessarily the answer. That's also the point. Because, yeah, you can sequence how many, I don't know the numbers that Jianming gives, but he is going to pay more for storage. He's going to pay more for analysis, and maybe whole exome sequencing is going to be cheaper for all those things and be enough depending on what you're trying to do. Or even you can do targeted sequencing. So actually, when the Center for Medical Genomics started, there were also the Common Disease Project, and they decided to go for genomes, and the Mendelians, we went for exomes. It ended up being more successful, the exome project, than the whole genome project. Autism ended up being more complex than they thought. The power calculations that I have initially about sequencing 100,000 autism cases, they thought it would be enough, then it wasn't enough, and all of those were whole genomes. Platforms, we already know them. No much secret here. So it's going to be the same type of library preparation. We're going to go through the sequencer, and you're going to get your FASTQ file. So there's no difference from anything we've done so far. You are familiar with the FASTQ file, so I'm not going to go through this. I'm going to stop here a second again, because we talk about the quality scores. I think it was the first day that we were talking about them. We only saw them so far, I think. Well, no, we've done BAM files too. So we see them, a couple of those, right? We saw them for the FastQC, or the FASTQ files, for the Align files. But they do exist also for variant calling. And it's the same type of a scale, right? They tell us how confident we are about the variant call we do, but also for the genotype, meaning is this a homozygous call, is this a heterozygous call? So alignment is a very crucial step. I'm going to just review this again, although we already done it. So we generally work with the FASTA file for our genome, but we need to build an index, right? A lot of aligners work with an index to have quick access to that information. So they don't have to scan the genome every time you need to align a read, right? So all aligners, so if you work with aligners, one of the first steps besides getting the genome is to produce an index of that genome. And every aligner produces a different type of indices. So whatever aligner you're going to choose is going to produce its own index files.
Francis, I have like a question. So if you are doing different kind of sequence, like 150 base pair versus 600 base pair, and you are actually aligning, do all these aligners use different indexing files for that? I mean, like,
Not for the sequencing you do. It's only based on your reference. So the aligner wants an index of your reference. So to quick access it.
no, that I understand.
So he's asking that will the alignment process be different for short read versus the long read? Is that what you want?
Or use of indexing.
So if you have a longer read,
Yeah.
will the index be longer, larger or something like that?
Well, it's going to depend on the aligner. So we have a specific aligners for long reads. So they're going to produce their own indices. And I don't know, I don't use them often to tell you if there's a big difference in the way they build it.
But the 150 and 600 will not.
Yeah, totally. No, BWA, which is the one we're going to be using, you can align in theory between 70 to 1,000 bases. So in that range, you're good.
What are we doing basically in indexing? What are we doing with our sequence?
So it's getting like a book, right? It's looking for fragments of sequence and creating an index of those fragments. So when you try to access and divide it by chromosomes and all that, so if you're trying to access a sequence, it's going to quickly know what it is in the genome.
So if you want to read one particular line, you don't have to scroll through the whole book.
Yep.
You just look at the index, is page पे इस जगह है, so something like that.
Open that. Whatever.
Because if we turn up again and again, it's going to take that much longer.
Yep.
Isn't it?
Yeah. So it seems, I mean, conceptually it seems easy that you say you have a sequence and I'm going to be able to align it to a genome. It is not, right? So first, it's very long. Aligners care about speed, basically. That's the most important thing, because if you work with whole genome sequences, it's going to be, you cannot be slow. You need to be fast. So the accuracy, you sacrifice a little bit of accuracy to be more faster. Another thing, our reference genome, or the one that we use the most, it may not be complete, right? I think we talked at some point about those issues.
Is the reference genome going to change because there's a lot of people with Pangenome or
Yeah.
like
Yes.
different people with different references
Yep.
and come with a consensus of what people are
That's ideal, but then all the tools are completely different. You throw away all the tools we have for new ones.
How far are we from a Pangenome? Like how long will it take when
There are already tools available and you can use them, but as you know, you also mentioned it in one of your slides, right? You were talking about most of the clinical data sometimes is AG19, not even 38, right? So databases, everything that is annotated sometimes is from even older versions of the reference, and then you need to transform it to the new one. So we have so much data now in the current reference genomes that it's easier to work with that than trying to do anything else. Right? But I don't know. I mean, they're available. We don't use them, or at least not in our group, right? We don't see anyone requesting them.
It appears that it would be more accurate. It could be more accurate.
That's the idea, yeah, right? That's the idea. So to being able to have all that information in a single reference. Because again, if you compare yourself to the current reference, 300 loss of function variants are going to be there. All the rearrangements, if you can analyze rearrangements, you're going to have thousands of those rearrangements. But that's one reference, right? We know that we are really complex if you look at those numbers.
Or is it possible to make a reference of like you already have like a million all over the world? You might have a million so-called healthy individuals.
Yeah, I don't know. I mean, that's a good question. It's something that I never thought.
Reference of that.
Yeah.
It would be easier, right?
Like accessing the variability at once, right? Yeah. I don't know. Yeah, if we can apply that information for alignment and vertical. Yeah. Well, we kind of use it to some extent.
Dr. Franz, the previous slide told that the repetitive regions are masked in the reference genome. So, what do you mean by
Sorry,
that?
with the AC, I cannot hear you well.
Previous slide.
Oh, the previous slide. Yes. Sorry. Yep.
You told that the reference genome presents complex regions such as repetitive elements, and the repetitive regions are usually masked to avoid them. So,
Yes. Yeah. But I mean, that's a problem you need to face, right? When you align. So not all of them are masked when you align. You mask them later if you want to, but during the alignment, you have what you have as a reference. We don't mask anything. So all the repetitive regions are going to be there, and they're complex. So there's another, right? We have the telomere to telomere genome in the middle before the pan genome, right? But still, we still work with AG38, which is from what, 2013?
Is it so difficult to change everything from there to there?
No, it's not, right? I mean, there are what we call it the shift overs. You shift from one to the other, but you still get weird stuff. I mean, it's not 100% correct, let's call it like this. You never get a complete shift over. Oh, that's what I was mentioning. Yeah, aligners prefer speed. So they make some assumptions, but the whole idea was that, so aligners need to compromise a little bit to deal with these situations. And another important thing, well, first, one way we're going to deal today is about the idea of soft clipping. So when the aligner maps something and the rest of the gene is not able, the rest of the read is not able to aligner, they soft clip it, meaning it's going to indicate only the part that it's been aligned, and the rest is going to be soft clipped, saying, okay, yes, the read maps here, but there's a section that I don't know where it goes. And the other problem of the current aligners is each read is aligned individually. And that's a problem, because they don't align, they're not necessarily is going to be aligned in the same way the next one. So you can be aligning in a region, the first time, the first read aligns in a way, second time, or another read which is not exactly the same, but they align in the same region, is going to align maybe slightly different. Because based on the algorithm, it thinks that's the best way, but every read is aligned individually. And this is a problem too, or it can be a problem. And I'll give you an example later. Oh, regarding aligners, yeah, again, this is a tiny representation, right? But these are probably popular ones. We're going to use today BWMM. This is a common type of algorithm that I'll mention briefly. Different languages. And we talk about this, being a splice aware or not, right? We talk about HiSat2, which is designed for transcriptomics because it's splice aware. The predecessor was bow tie 2, and then they went to HiSat 2, but bow tie 2 we still use it for some applications. They're based on a slightly different algorithm, but again, they are not that different.
So when we use genomic data, then we use different algorithms.
Yeah, you can use different algorithms. The goal is going to be the same, but you can use different algorithms. Of course, there are aligners like Minimap2, which is designed specifically for long reads. We're talking about pangenome references. So this one is designed for pangenomes. It uses a completely different idea, the graph-based mathematical approaches. Most of those are going to be command line interfaces. They are more flexible and easy to use with big data.
CLC has got a Java interface.
Oh, it has also a Java command line to use it? Okay.
No, the CLC I said has a Java interface.
Oh, it's written in Java, yeah. But then also some commercial, and I don't know if there's any free ones, but you can use graphical interfaces too for alignment.
Is there any advantage of using the commercial one?
Well, I don't know if I have changed. It's been a long time that I don't look at benchmarks, and I'm blocking the name. Oh, NovoAlign. There's one called NovoAlign, which is commercial. That always shows up as a best aligner. Even better than what we use as standard. But of course, you need to pay for that. So I would say the most common standard nowadays for alignment for genomic analysis is going to be BWA.
Novo line was written by Jim.
Sorry, what?
Novo line was written by Jim.
No.
What was Jim's alignment?
I don't remember. It was designed for 454 data. This is the basic idea of HiSat2, also BWA. I don't think it's worth it to go much into this. But the main idea is they take a fragment of your read. It doesn't take the whole read, right? So for a speed and compromise, right, it takes a fragment, looks where can go that read, it maps that, and then tries to extend it. And see, okay, yes, this short fragment goes here. Can I extend the read and keep mapping? Yes, good. Then I'm going to say this is the place. If not, I'm going to look for a different spot. So just to give you an idea, a general idea how it works. They call it a seed. They find a seed and they extend. Seed and extension. The SAM file, we already saw it. I don't know. You have questions about this? But every line in your BAM file represents a read, an alignment, a read being aligned, and there's information about where it maps. We even look at this is called the flag, or the SAM flag, that it tells us what type of read it is and how it aligns. The position where it aligns. Oh, this is a terrible mapping score. We already know that. This is the cigar string that it tells us how it aligns. It's exactly the same slide, and I remember there was soft clipping. Yeah. So this is the idea of soft clipping. 47 match perfectly. 54 is being soft clipped of that read that doesn't know where it goes. So the first 47 bases matches, the rest of the read doesn't. So I'm going to soft clip it. And it gives me a very low mapping score, so we don't worry about it. SAM tools is the tool for manipulating alignments, and I think we're going to see probably in the Jupyter notebook again how we can use it. So sequencers are also a problem. They produce errors. PCR produces errors. So the main goal, or one of the goals of the variant callers, or variant calling, is to distinguish errors from non-errors, right? So here I'm just giving an example. So this we're going to be more or less confident that it's a real deletion. Here we're going to be more or less confident that it's a single nucleotide variant. And here we're more or less confident that there are sequencing errors. And there are two reasons. But we're going to go for one. What do you think makes us think, or what is the main reason that makes us think this is an error?
We can't find others like it.
You said because it's at the end? That's an interesting answer, yes. And?
The nucleotide is
The same nucleotide is being changed in all reads.
generally like only C or C.
Yeah, you're going for the second main reason. But yeah, that's another point, right? But there's one basic one. There's even more basic one.
It's not very frequent.
So the reads are not
Very less,
aligned
very less.
sequence.
So yeah, the coverage, right? The coverage that we have of the answer, right? So here we see 3 times, so the deletion and 3 times the reference base. So half and a half, something expected, a heterozygous call. Here we only see it 1 time out of all a few. I know this is very, very shallow sequencing, but if you have more, you can be more confident that this is going to be sequencing error. The other one is the allele. If you analyze the whole allele, you're going to see the C and A. They only appear 1 time when you're expecting the CC combination. Except if it is somehow it has 3 copies, right? We don't know if it has 3 copies, but we can say that. So I'm going to skip this because we already answered. So the main answer is going to be, we're going to base our, or how confident we are about a call is mainly going to be about, the main reason is going to be based on the coverage that we have. The more coverage, the more confident we're going to be about the calls. But that's a good reason too that we're going to go through that because you said it's at the end. We'll talk about that too. I don't need to explain this again, right? We talk about coverage, what it means. So it's how many times we see every base. We also sometimes talk about the breadth. And I think I showed this probably the first day. So I'm not going to stop here. Visualizing, again, the idea of visualizing everything we do is really important. Dr. Amin also mentioned it probably. I don't know if we talk about it, but at least I think it sounds familiar that you mentioned it. IGV as a good tool for visualization. We are not going to use it because it's graphical. I mean, we don't have time for this, but it's easy to install in any laptop and you can visualize your analysis. Not only for variant calling, you can use transcriptomics on this too. You can see how you read, they splice across exons. That could be a good way to look if you are detecting a transcript with a specific exon or not. You can visualize them too.
You can also visualize splice site variants.
Yeah, exactly. That's what I'm trying to say. The splice variants, right? So you can visualize them using IGV. If you are missing an exon or not in your transcript, it's a good way to do it. Or you mean single nucleic variant that breaks the splicing? Yeah. That's a good point. Yeah, if you find a variant in the splice site and you want to test later with transcriptomics if that variant breaks another splicing, that's another way to do it. Yeah. So did I miss something here? Okay, so if you have a call and you don't know if it's real or not, you can visualize it, and you make a good point earlier. So why we think this is a misalign is because it's at the end of the reads. So aligners have issues at the end of the read. Sometimes, this is what I was trying to say earlier, they're aligned individually. So this, when it was aligned, it was fine. Said, okay, there's nothing here. But then I'm aligning another read that covers the same spot, and I'm saying there is a variant. They are aligned independently, and statistically, this looks like it was the best position for that alignment, and the way to align it was correct. In this case, you need to be careful about variance at the end of the reads, because maybe decide, okay, if I create a gap here or not, it's going to look better. It's most probabilistic thing, right? So it's quite common that, or quite common, if some errors can happen because misalignment at the end of the reads. I'm not going to say it's common, but it can happen. So if the only reason, what I'm trying to say too is, if the only reason that you have to believe that this is real is based on 2 reads and they are at the end, you need to be suspicious. So this is another problem that we know the aligners encounter, which is when you have some repetitive regions, it's going to have a really hard time to decide how it goes, right? So it may create gaps or not gaps or...
This was from Iron Current, or this data or from like Illumina?
I don't even remember where I got this image. Oh, I'm telling you, it's from a paper, it seems, of somewhere. Yeah, it's a paper. So now we need to go from this alignments to a list of variants, right? And giving the genotypes for every variant and how confident we are. So that's the main role we're going to do with the variant caller, right? So we go from some alignment to a list of variants. So that's going to be the next step in our process. And also trying to make an effort if we want to classify them, the current models of calling variants. So again, probabilistic method, we're assigning a probability of being some algorithms. We use an algorithm to assign a probability of a call being correct or not. So these probably are the two popular ones. JTK definitely is one popular one, and it's the one we're going to use. Probably the most popular one, but there exist others. And you can even combine them. There's ways that actually our pipelines combine more than one to produce the final list. And of course, we're at the time of machine learning, and they do exist too. Google created its own. I mean, a lot of the different aligners that use machine learning approaches. And
You can give the slide over to him.
to him? No. I'm going to feel bad. And the graph base, what we said before, right? So the Genomic project is going to we're going to need completely different aligners. Because we don't work with a single reference. We're going to work with multiple references. So that's a completely different paradigm. And we're going to completely need a different type of aligners.
So but the Panjab will it not make a consensus kind of decision?
No. It creates a graph too.
So it will align against each of those demotes then?
Kind of. Yes, it creates some sort. There are common regions that are going to have less complexity, other regions are going to have more complexity. So they create some graph approach. Again, I'm not good at it. I never use it. I'm not that familiar. So what about variant callings? You have a long list too. You have a few of them, and they're going to be based depending on your data, right? So we have, depending on what you want to do with data. If you work for single nucleotide variants, you're going to use some types. If you want to work with a structural variants, you're going to use a different type. And if they're short or long reads. So they're quite specialized depending on the type of data that you have and your goals.
They're all free to use, or like there are some other like commercial or like
I don't remember which ones they were included here. I took it from another paper. And I don't think this is the complete list. So actually I selected a few because I wanted to give some examples of pangenomics. Which one it is? I don't know if I remember which one it is. Or for example, low frequency. This one is for that in theory was designed to detect very low frequency variants. Like if you have really low somatic variants, or if you work with if you CRISPR screenings, or for CRISPRs, you create some sort of mutation, you know it's going to be in a low frequency. You can use different type of variant callers. Because again, JTK is going to assume that you're working with a human genome, and the expectation is going to be that if he's a heterozygous call, it's going to be 50% one call and 50% the other, right? So some of them make assumptions about the expectations of the data. So that way I don't have to pass every step. The JTK, that's the one we're going to be using. It was created in Broad. It's a command line tool. You need to know that. So we're going to go to the Jupyter notebook. We're going to use it to use command line. So we are not going to work with R today. Well, a little bit, but not much. But basically, it's going to be command line. So if you feel comfortable with the terminal, you can go to the terminal and use it. JTK is a set of tools. That's also important to know. It comes with, it's not just a variant caller, it does other things too. And you can also use it for somatic calling. There's a specialized tool within the toolkit that is designed for somatic calling. This is the website. There's really good documentation in the website. And they have pipelines and what they call the best practices for variant, not the best practices in general for variant calling, but the best practices of JTK variant calling. This is a graph they use to explain the best practices, so all the steps, but I'm going to simplify these in a few global ideas. And I go into the details of 2 or 3 of them to know, but the main goal is going to be to create a file with your list of variants. That's the main goal. We've seen some of this already, but these are going to be the steps I'm going to mention. I'm not going to mention the first one, of course, because you already know FastQC, we already use it. But did you notice something between these 2 steps? We are not trimming anything. We are not going to touch the data, especially today. There are ways you can do some trimming. For that, it's better if you check the JTK documentation. But we are not going to trim anything. We are not going to touch, or we are not going to modify how the read looks like in the sense of quality.
Is it specific when we talk about whole genome, that trimming is always with the transcriptomic data?
No, it's not necessarily only for transcriptomics. There's other applications you could do trimming. For variant calling, generally you're probably not going to do trimming, because trimming is going to modify the quality of your read to some extent. And we want to keep some of those bad quality scores too in the process. So we are not going to touch it. I mean, you could do trimming. So I'm not saying you don't have to do trimming. So in JTK, there's some documentations of ways that you could do trimming to your data. And there's tools for that too.
But if your variant is in the trim one,
Yeah, if you remove that section, you may be missing variants too. And ideally, we want to cover as much as we can and not as much as we could. BWA is going to be our aligner. We're going to do one thing that we haven't done for transcriptomics, which is removing PCR duplication. PCR duplication is not giving us more information. It's just adding biases to our estimations, nothing else. So we want to removing. There are 2 types of PCR. I call it PCR duplication. Let's call it duplication in general, because it can come from the PCR, but it can come also from the sequencing machine. And those are, we call it optical duplications. But there can be a couple of sources where duplication can happen. We're going to do one thing that is called base quality recalibration. And that's why we don't trim. We're going to go back to, we're going to say sequencing machines are giving us not the correct quality scores. We're going to recalibrate them to something that we think is more correct. For the problem of the call is at the end of the read, for example, right? Or because they are homopolymers, let me readjust the alignment. So JTK, not anymore, but I want to talk about this because it's interesting. I think the latest version, definitely, I think from version 4, they don't do realignment or reassembly of your reads. They change the way they do it. But it was an interesting problem to solve, what we said, right? So calls at the end of the read, they may be misaligned, right? You could solve it using what they call local realignment. So I see some variant. Can I get a locally, take into account all the reads, can I locally do a better job that the aligner did?
so local realignment means that it will look at the neighboring read
Yep.
and say that, okay, 10, 20 reads at this are showing this and only 2 are showing, I correct it to that.
Yep. But again, this is not happening anymore. It is not part of the process in the JTK4. That's the one we're going to use today.
So do we get the information in, suppose in 30 reads, these many are of this type and
Yep, that's part of the BCF format that I'm going to talk in a second. But yes, you're going to get that information. Oh, and of course, we're going to do variant calling and genotype calling in our last step. Or that's, let's say, the GTK alignment process of variant calling. I pretty much mentioned all of them. So I was trying to say that, so when you have some sort of duplication of sequences that you know that a stars and n, they are more or less identical in sequencing, those most probably became part of a PCR duplication. They are not giving you information. They're creating a bias when you try to estimate how many times you see a base, right? So if there is a base here and you're trying to estimate how many times you see, this PCR duplication is creating a bias of the count. So you want to remove them. We don't do this in transcriptomics. It would be great if we could, and probably there are methods that we could do it. PCR duplication can exist in transcriptomics. It can be less if you use less cycles of PCR, but it could exist. But you're working in such a small space, which is the exome or the transcriptome, compared with genomics, and duplication is expected because you are sequencing the same transcript multiple times. The optical duplication happens during, Bonnie probably can explain you better. There's some breach process during the sequencing and the spots of the flow cell. And during that process of breaching and duplicating sequence on spot, you create PCR duplicates too. I don't know if there's other reasons for optical duplicates in sequencing.
Yeah, so optical duplicate is that, and then so there are basically what happens in the linear sequencing. It's hard to separate between optical duplicates and PCR duplicates. Okay, so there's only blanket term which we call PCR duplicates. Some of those reads come from the sequencing platform, what Hansel just explained, and the other part comes from the library file. So together, whatever we are what we are saying is actually coming from.
Yeah, so the tool can even somehow split between what considers an optical or not. We just put it together and we go.
So, and each library type, there are some callable limits, like for the variant calling, when we are doing whole genome analysis, usually we make PCR-free library, and the number what we are looking for is around 10-ish. If it is more than that, and we don't expect it to be more than 20%, when it's high, but if something around 10%, we like it.
So today I'm saying a lot of so. I mentioned the base quality scores recalibration. So the idea comes from if we observe a variant in the population that we know it existed in somewhere else, most probably if we see in our sample, it may be real too, and not an artifact. So that's the global idea, right? So what it does is, so we have a known set of variants that we can use. It's going to look about those mismatches. If it's present on the reference, we're going to assume it's real. And if it's not present, I'm going to recalculate the quality scores, take into account more things. The position in the read, whatever's around it, and taking the original quality scores also in consideration to calculate a new quality score. So we're going to be modifying whatever the aligner tells us about the quality score of the base to something different through this process, by taking into account more information. Can you see a problem here? Or a problem, or you think the problem is going to be whatever we use as a reference. It's not necessarily correct, right? Sure, but I mean, I'll give you an example of probably that it's not necessarily correct. So one of those is dbSNP. So all these IDs are from dbSNP. So dbSNP also has errors, I'm sure. It's not going to be a big deal, but they exist, so you need to be aware. The realignment, reassembly idea, so here I was trying to give an example of before and after realignment. So here is the problem, right? Before realignment, you're going to detect two SNPs that already are present on a database, so you assume they're real. But when you do the local realignment for that indel, you realize that they're gone, right? So are these reals or artifacts that somebody else published as correct when they are not part of they actually don't exist and it's a problem of the alignment?
But in the second one on the right,
Yep.
there is a deletion of code request, like here.
So here, so this would be like
The other RSID of that. No, there's 3, 4, yeah.
oh yeah. So this represents the indel, yeah.
So that was already there on that also.
Oh yeah, it's present but not called to some extent, I'm assuming, because there's no support, or was called probably, yeah.
So it improved after you did the
local realignment. But again, this is not happening. So local realignment looks, so the alignment failed because basically it takes every read at the time and it doesn't have information about the rest of reads. Local realignment looks, okay, there's a situation here, can I improve it somehow? Are these correctly aligned or not? And it comes with the conclusion that if probably this is more real than this local situation. again, no, it's not happening anymore in JTK4. It uses a different approach. But I thought,
Why did they decide to do away with this?
I'm assuming they consider the new approach better. I don't know the details. But I mean, we've been using GATK3 for a long, long time, right? During the teens is what we use the most. I don't know when we started to use GATK4, but we've done a lot of analysis following these steps.
So that, but the previous slide is just showing the known set of variants. The T was the known set of variants.
Yep, all were known.
Okay.
All were known. So they passed the previous filter of base quality recalibration, but not the local realignment.
So what we are seeing in DBSNV, some of them may actually be false calls.
That's what I'm assuming, yeah. Again, I'm not an expert on those databases to tell you how many are wrong or not. Or
So are they correcting it when they have been proved experimentally? No, this is not a true.
I don't know. I have no clue.
Because we rely so heavily on that.
Or maybe they exist. In this example, was just not there, right? But because existed in the database, we call it, yeah, this is fine. We see it in the database. Maybe they were real, but not in this sample. Definitely not in this sample.
And these false calls were picked up on the IgG. After looking them on IgG, they were, I think,
Well, there were examples of this situation more than anything else. We don't go into that deep of the detail, right? We generally, I mean, not in production settings like we are, right? We trust whatever the aligner do and whatever the JTK tell us, and we worry about other things more than going back into the detail. Here I'm pushing or giving the idea or giving examples of what happens in the background.
So are they
You are not going to see all that, by the way.
commercial pipelines like Dragon also using JTK, or they have got different algorithms?
They change, right? I mean, the first idea was to use JTK. Yeah. So they use something, their own color right now. I don't know, I haven't used. Bonnie, do you know what color Dragon uses? They have their own, right?
Yeah, they have their own. They are actually Dragon servers actually, and they are GPU accelerated, so they have their own product.
So a Dragon server, what it is instead of having a computer with the software is inside of the chip itself. So that's why it's so fast. They have a name for those chips. I forgot how they call them. No, they have a specific name. PG, I don't know. It's been used in other settings, not only. So Dragon chip is not new. It's been used for other things. So the last steps, basically, if you have multiple samples, we have a specific calls for every sample independently, but you can do another thing. So especially if it's a big cohort, you can use that information of the cohort to improve your calls. Because you're expecting that cohort is going to be more or less similar. So you're expecting similar calls. So you can even improve your final set of calls using the cohort information. If you're working with less than, I don't remember, 50, there's some sort of cutoff that we generally say that we consider that for good for joint calling, cohorts, or just individual calling. In any case, if you you have multiple samples and you call them separately, what you want is to put them in a single file. So that's generally what we do. Even if you have 3 samples, you're going to end up with a single final call after some filtering that we can do. And we will do some filtering.
This final BCM merging, this is only for cohorts, or like you do it for?
No, we do it even with 3 samples.
So for a trio also you will have a final BCM?
Yeah, with a single one, yeah. We generally do that. I mean, you can keep them separated if you want, but it's simpler to work with a single file.
So but it will have information, for example, if you're working with a trio.
Yep.
So in the same BCM, you will have variants of the father, mother, and the child.
Yep.
In one BCM, and
Yep.
mark which is coming from there.
Yes, so, and we'll see it in the practice. There's a little bit of a complication for that, but yes, that's how we keep it.
But how do you know which was coming from where?
You give the genotype. So there's going to be columns for genotype for the mother, father, and proband, for example.
Based on HLA or which one?
No, the genotype of that position. So if you see a variant in one of the 3, you call that, it's going to be present in the final file, and then you're going to say it is.
So you sort by positions then?
Yeah, your final list is going to be sorted by chromosome position, and you're going to have the genotype of every individual at the end for that specific position. Oh yeah, so the next thing, so the BCF is our final product, is variant calling file. That's the name we give it, BCFs. You can have them like BAM files. We have some BAM. You can have them compressed, and then we call them BCF with a B. I'm really bad differentiating between the two, but yes, V and B, BCF files are the type. There are specific tools to work with it. There are some columns, the minimum number of columns, basically chromosome positions, the reference allele, the alternative allele, the quality of that call, if it passes filters or not, there's information about that. And the info field. Info field can be really big. With, like, also exists in the BAM. We can have a lot of information about the alignment, and it can change depending on the variant calling or what you do with that file. But yeah, we can have a pretty big info field too, with information about the variant. So this is how it can may look like for a simple sample. We're going to have a bunch of header lines. But after that, every line represents a call or a variant. Chromosome position. You can even have ID information there, like DB SNP IDs, if you do that or annotate them somehow. Reference, alternative base. The quality scores we've been mentioning all the time. So you notice that it's a different scale from sequencing that goes up to 40, alignment goes up to 60 normally with the things we do. Even you can get 90 for a call. If it passes another filter, the info field that I mentioned can be really big. Here I'm just giving a quick example. So this is the depth of sequencing, and this is the allele frequency that you observe. So what we're saying, how many times you see it? You see it 56% of the time you see the alternative. And the format for the sample that is going to come later. You can have multiple samples here. This is the genotype. The number of times you see, in this case, is the genotype. It means, are you familiar with this annotation? Being 00 or being 01 or 11? No? Okay, sorry. So I need to probably stop here. I think I have. Yes, I do have what it means. So we generally use 0 for the reference. So if we have, so we have two copies, right? So of the information. So if we say 00, it means it's homozygous reference. If we say 01, it's heterozygous. If we say 11, it's homozygous alternative. If you are working with a lot of samples, you can be in situations that you have even two present there, right? Because there's some sample that may have a third allele that is not present in the others. It can happen, I guess. Especially if you work with indels, and the indels are weird.
In the sample 1 means heterozygous. So what is the other?
Yes, so this is reference allele, the number of times you see the reference allele, the number of times you see the alternative. So I'm assuming these should give you the 56 if I didn't made up the numbers, which I'm not sure, because this is just one thing that I put here. I probably put some numbers on, but I'm assuming if you do this division, it may give you the 56, I guess. So
What about genes where there are copy number variations?
Well, that's a different problem. The assumption here, or maybe the first pass analysis, is that we did two copies, right? But yes.
Some genes which might have as many as 6 copies, 8 copies. So how do you do that?
Well, that's you need to use something different, right? Or estimate the number of copies in a different way. But for variant calling, the caller is going to assume that you only have two copies. Yes, we do it. We do it with example data. And generally, the copy number situation that we're talking about now is generally solved based on depth of sequencing. It is the basic information you have, or the only information that you have.
No, but then you'll have to have a mean of a number.
Yeah. Yes. But there are callers for copy number variation even in exon. So there are specific tools for copy number variation in exon sequencing. They generally use the concept of that idea, or the basic concept behind it is based on depth. Right? You expect that all exons should have the same depth for sequencing, and suddenly one or a whole gene changes the depth of sequence. You're giving a probability of having a copy number situation. That is really common when we work with somatic calling. When you work with tumors are really unstable, and copy number variation is something that you need to analyze when you work with tumors. 14 is the number of times that you see G. 18 is the number of times that you see A. So there are 14 reads that align in this, or there are a total of 32, right? 14 plus 18 aligning. 14C and a G, 18 they see A on that situation. Sorry, my throat is itchy now.
That is 32.
32 is the def, again, is repeated here. It's telling you based on this. And 99, again, quality, genotype quality. How confident are we this is the right call? All this information I'm explaining, I don't know if we're going to see it in the hands-on, but generally it's in the header. It's explained. The header are really long, telling you all the information that I'm just telling now verbally about your file. Even myself, there's so many codes for these things, sometimes I get confused. I don't know them all on top of my head. Oh, here, I did it. So this is a header. I wasn't sure if I include this. So if you're curious about the DP, the depth of sequencing, it tells you approximate rate of read depth, and GAQ, genotype quality, and so on. You have pretty much a lot of fields that you can have there. Probabilities in a different type of approaches, you can have them too. We also talk about this, Dr. Amit talked about this. Yeah, we have our calls, but we don't know what they do. So we need more information. So we're going to add annotation to it. There are different types. There are different tools. Here I'm mentioning 3. So this is the 3 we used in our pipelines. We even use more than one, which is for really specific things, which doesn't make much sense, but we do.
But the outputs would be different.
Yeah, but we have to merge them, which is an extra work we do in it. But I mean, we build a pipeline, but we merge them, yeah. For specific reasons, but we're going to use today BEP. It was a pain to make it work in our system, really a pain to make a BEP to work. That's one of the reasons, now I'm going to get a bit technical when we talk about content environments, that what I did here is not ideal, which is I created a content environment for every project we've done. From bulk RNA-seq to single cell to variant calling is a single gigantic environment. Don't do that. Divide it by your projects or type of analysis you're going to do. Because then you encounter that the variant effect predictor doesn't work. Or it was installing a super old version to make it compatible with the rest. It worked for a day and it crashed after that and it couldn't make it work. So the way again, I'm going to get technical, but just to give you an idea of solutions, if you need to work with solutions, to make it work, we use a container. You know the Docker containers we talk about? So this is in a container by itself inside of our content environment to make it work. It wasn't another way because we couldn't make it work to make it compatible with all the other tools that were available. So again, so for annotation types, we do a lot of different things. I'm mentioning here a few, but one of the most important ones is what is the consequence? What's the impact of this, right? It is a loss of function call. Loss of functions, I think I probably mentioned later. So let's go through the next slide so I explain what type of annotations we can get. And the main goal, right, or one of the main goals, especially in rare variant systems, but probably in anything or a lot of type of analysis you want to do, the reason of annotating is because you want to do some cascade of filterings, right, depending on your project. But generally you want to do some cascade of filterings to explain your phenotype, whatever phenotype is the one that you're studying. Yeah, we can start with, I think on average, I think mentioned we have a lot, between 3 and 5 million variants among us. Not all of them are very important. You can classify them based on the consequences and say, of course, a synonymous variant is not going to be important for my setting. I'm going to remove those, and so on, and so on, and so on, and so on, until you probably get one or more, depending on your setting again. But if you're working with a rare variant, the expectation is this one explains your phenotype. So that's what you're trying to get. So the impact, we were talking classification of the variants based on the impact. And I talk about loss of function, and I realized I maybe didn't explain what loss of function means. So BEP classify them in high, moderate, low, and modifier. And these are the types of loss of functions that I was trying to explain. If you find a stop codon in the middle of an exon, you know that protein is not going to be functional. Or it's going to be highly not, most probably it's not going to be functional. maybe functionally if you're lucky and it's the last exon of your protein and it doesn't do anything important and it's probably something that, okay, you can have situations maybe that the protein survives somehow with a stop codon maybe at the very end, right? But yeah, you have other things, like if you have an indel that changes the frame shift, you know that it's going to have a high impact consequence, right? One of the biggest problems, missense variant, you know there is a change of an amino acid. That's going to be hard to figure out if that change of amino acid does anything. For that we use other type of annotations, but they could be interesting, definitely. And the low impact. We don't know what a synonymous variant may, or we're going to assume generally that a synonymous variant does nothing, but they may do something. And we even have cases there are cases, for example, that modify another variant, right? So,
What if they are at the splice site?
well, yeah, if it's a splice site, we can even, I think, so a splice donor variant.
I mean, a synonymous variant which is lying close to the splice site.
Yep.
It might change the.
I'm sure. So there's never like a, so you can never be completely sure that it's not doing anything, right? We talk about the missense variant. So there are tools for that to they try to make an effort of classify them as pathogenic or likely pathogenic or not pathogenic. Sorry, like pathogenic, likely. They can. I shouldn't use that word, but it depends on different. They have different names for everything. We can say it's a deleterious variant or a tolerated variant, a benign variant, however you want to call it. So there are multiple tools, and you're not going to be safe only using one. That's probably the message that I want to say, is that one is not going to solve all your problems. There are discordances between them. Some of them are based, like meta-SVM, meta means some sort of meta-analysis. You're going to put together more than one of those estimates and try to give you an answer. Others are based on, they're based on different systems. I'm not going to go all through them, but the main message is that one is not is enough to give you an answer. And generally we use, I think in the current pipeline we use 7, and the one that we use. 7? I think it's probably some number around those numbers. That's the one that we generally use for missense variants. For intronic variants, we use another type, but also they exist for intronic variants that try to give you an estimate of the probability of that variant affecting the protein. I don't know, the popularity of those, if I need to choose one, we work a lot between CAT scores and meta-SVM and Rebel. So Rebel, meta-scores, and meta-SVM, they were the 3 that we use a lot at the end of the CMG project. We're the ones that we trust the most. But I mean, you cannot trust a single one.
Rebel is also a meta tool. I mean, it combines.
Yep. Another important one, right? For rare variants, I think you heard that before. The expectation that a disease is being caused by a common variant is probably not. It's unlikely, right? Especially if you work with rare variants, you expect that the variant that you're studying is going to be less than the prevalence of the disease, right? Significantly less, but because a lot of diseases are not explained by just a single variant. There's going to be multiple variants that explain the disease, so for sure, your variant needs to be less than the prevalence of the disease. Assuming full penetrance. The penetrance is a different problem too, right? Then we have ways to annotate for frequencies, and there are different databases for that. Being Nomad, a popular one, and you have it for genomes and exomes, and they're being combined in a single one now. Conservation, that's another way you can annotate. If your variant is being conserved in a lot of a species, you know, or the amino acid is conserved in a lot of a species, you know that amino acid, if it changes, it may be an important change that you need to study. If it's an amino acid that changes in a different species, you can think, okay, yeah, I mean, it happened before, it may not be. If it happens now, it may not tell us much about it. There's measurements about intolerance. So if you're working with a gene that has, in the normal population, has a lot of variance, you can assume, okay, a few more, it may not change. It may tolerate variance compared with another gene. So that's another thing you can look at it. There are different databases that already include variants and can give you information about it, like Cosmic, I think we talk about it, which is for cancer. These two are for clinical variances. OMIM is not for variants, but it's for genes. How many people, I don't know, I don't know how many people here is a clinician and uses these things, but this is for variants and this is for disease, OMIM. Are you familiar with OMIM? No, it's for Mendelian. Yeah, it's proper. Oh, okay. OMIM. Yep. Cosmic. I said Cosmic is for cancer. So there's a lot of databases that you can.
CLIPAR and OMIM, a lot of clinicians use now.
Yeah, totally. The problem of annotation tools is always you need to update them because those databases they update. So the tool you use need to be updated too, and so on. It's really hard to keep track. This wasn't a slide that Bonnie showed, and I want to show it for a different reason. What year was 2011? Whole genome sequence of siblings. I don't think they were twins. And this was the process of filtering, right? You start with 6 billion bases, and you keep going the cascade of filtering up to you get, here's the 3 candidate genes, and there was only one that was shared between the siblings, and both of them were affected. So they decided, okay, this is the most likely gene that causes the phenotype.
What was the disease?
I don't even remember. But it was a coding variant. That I remember. So could have done whole exome sequence and solved the same problem? Yes. Yeah, the last slides, I think I did only a few more. Let me know if you have questions. The problem of calling somatic calls is different than calling germlines. We use a specific callers, which is part of JATK, that is called mutic. Oh, there are more. This is not the only one. You have other options.
But the somatic variant can also occur at different times. It could be just post-zygotic, it could be much later.
Yep. So here we are kind of representing the evolution of a tumor, and you have a problem of clonality also in the tumors, right? So if you have your analysis, you do your analysis here in time, and you can imagine, I'm going to come this in time, I have the tumor, I'm going to do the analysis at this time point, you're going to observe, and you may have different clones in there, meaning different clones having different variants. So now the frequency that you see a variant changes, and the number of variants that you can have, they can change too. So this is a hard problem to solve. So tumor clonality is the main issues, right? Germline callers are not going to work with this. The copy number variation that we mentioned earlier is also a big problem. Purity is a problem too. I don't know how many times pathologist comes or send samples and they tell us, or somebody that works at the Department of Pathology and tells me, oh, this is really pure. And then when we do the analysis, it's not, right? And try to explain that it's not. There must be cells in your tumor that are normal. And the data tells us that it's not pure.
There are some cells in tumors, immune cells.
Yeah, of course. They're infiltration, right? Yep. Oh, here I'm just trying to explain the problem. But yeah, you got the problem that purity and all the factors we explained earlier, it's always going to be a problem. And as such, you need to go deeper in sequencing.
So for tumors, you do like exome with a high coverage, or you do a panel?
Oh, in the clinical setting, they may use panels for research. I don't know. I'm not familiar with clinical settings so much. I don't know what they do, but I'm assuming they can do both, probably.
No, I'm asking that for tumors because you need high coverage to get the...
Oh, well, they do. We modify the whole exome pipeline, for example. We spike in to cover 500 genes in a higher coverage.
Those are sequenced at 600x coverage on a copy. So those 500 genes are sequenced at 600x on a copy.
So the rest of the genes you will do at
150x coverage.
and then the others you will do at a higher
500x, 600x.
So how do you come to know that, I mean, the data is from, not from tumor, but from the normal, I mean, the surrounding cells?
Oh, we use frequency. We look across all the heterozygous calls to calculate that. So, based on expectations that 50 and 50. So based on the frequency of the alternative allele across the whole data set. So in theory, if you should expect at least 20 in an ideal world, right? So a pure 100% pure tumor, the head call should be 50%. Any deviation from that may be caused, of course, by biases on the sequencing, but also by not being pure. And you can analyze all the sets, and you could assume that on average, you're going to get a good estimate of the purity. We observe purities between 40 and 60, and that's totally normal. Below 30%, it can be problematic. You're going to be missing a lot of calls. Again, it depends on the, all depending on the coverage, right? We have the problem of the coverage here. But let's say in a standard setting, we generally say, again, I'm not a person that works a lot on tumors to tell you what is the best approaches for this. But I would say, I think what we observe is between 40 and 60 on purity. It's totally normal. Below 30% starts to be problematic. Yeah, so about long reads. Why we use them, right? So they have their own applications, especially dealing with the structural variants. Whole exome sequence is going to be a problem for these things, right? We are not going to solve structural variants with whole exome sequence. If you do whole genome and you short reads, they are short, and ideally you want to cover at least the breaking points or go over the lead or over the structural variants to solve it, because it's going to be, it's a complicated problem if you work with short reads. Tandem repeats, of course, another problem. We can use them for phasing. That's if you care about phasing, right? So you can imagine that in the same gene you have two calls, and they are far apart from each other. You don't know if it comes from the same allele or from different alleles. If both of them are stop codons, I'm making this up, or they are loss of function calls, if they come from the same allele, you may think, okay, that's fine because you have an allele that produces a protein that is functional. But if it's in a compound heterozygous situation, that's how we call it, then you have a problem because they come from different alleles. A way to solve these situations of phasing those calls is with long reads, because you can cover both of them and you know if they come from the same allele or from different alleles. Of course, full transcript sequencing is another application. And probably there are others, of course. Assembly of the genome. And that's it. And my favorite, I wanted to finish with my favorite quote. So yeah, talk to your analyst if you are not the analyst. It's always good. Designing the experiment in advance is always good. Okay. Oh, we still have an hour before lunch. Should I start? Yeah. So if you have questions, that's a good time. I need 2 or 3 minutes. trying to log in? Here. Are we waiting for somebody else, or we can start? It seems that I'm missing people. I'm not sure. Let's start. We're going to go to the A5 folder, variant calling, and there are two subfolders. There are two data sets that you could practice for variant calling. We're going to do the trio one. So we're going to open the trio WES variant calling. And the notebook we're going to be opening is the variant calling JATK. But before that, just show you that three folders. So each one of them is a different sample. Inside of that folder, there's an aligned folder. Inside of an aligned folder, that is the FASTQ files. I also have the tendency to make them more or less independent. So if one day you come back, I'm explaining the same thing in several Jupyter notebooks. You've seen these before. So I don't think I need to explain how. Oh, yes, I need to explain again. It says this is going to be running commands in the terminal. This is not R. Okay. We are using the Python 3 kernel, and this thing here is to tell that we are running a command line, a bash command. You could do all this, I think I said a lot of times this, opening a terminal and running the commands in the terminal. It's the same thing. There's no difference. And even some people tells me I shouldn't do this. I should teach you how to use a terminal instead of using a Jupyter notebook. I don't know. I like the notebooks because you have the text. So all this is just to show you how a variable works in Bash and how you assign them, and how you use them. We've seen this before. The idea of how we run commands. Generally, we go to the folder where the software is. You run that software. We don't need to do that. Because everything is in the path variable, right? You all remember this, right? If not, tell me no. How to get help. Yeah, here is the idea of the path. And why in Conda we don't have to. So I like to have it that in even if we just mentioned this not long ago. In case you go back to a single notebook and then you don't remember why we're doing things, you always have it on top. Oh. It's only me who lost connection to the Wi-Fi?
It's me too.
Okay. Well, more or less, you see, I'm just listing the steps of the analysis. And I also put links in the notebooks how to get more information too. So here you have the JTK best practices. There's a lot of documentation. There's a lot of FAQs or people making comments, and there's a lot of answers for common problems too in the right order. So we're going to do a FastQC. We're going to align them. We're going to mark duplicates, and that's something that I didn't say in the slides. We don't remove the duplicates. You could remove them. We just mark them and say, this is a duplicate. Don't use it. Base quality scores recalibrations. We're going to do variant calling with this tool that is called haplotype caller. All this is JTK, by the way, but they have tools. JTK is the tool set. We're using one tool inside of the tool set that is called haplotype caller. We're going to join genotype all three samples to have a single file. We're going to have some steps of filtering to remove variants that we consider probably are not real. And we're going to annotate them with BEP. So everybody's back to the notebook. I think I mentioned this, but just in case, because you're going to see them a lot, I think, in this notebook. The command line expects your codes in a single line. Normally, there's even exception for that. But a way to break the line is using the // symbol. And it assumes, it says, wait, don't execute it. Let me keep writing. Wait, not execute it. Let me keep writing. Now you can execute the line. It's a way to break the line and make them more readable. You're going to see this often I think you see this often in this notebook. And what we said about the samples, another thing that we don't teach enough maybe is how to be organized in genomics, because you're going to get a lot of data, and it's hard to be organized. I know some people come from different fields, and I assume things that people know, and I'm realizing that it's not the case. And we create a lot of folders with long names, and then some folders, and it's good to be organized, so you need to be organized. Do we need to do this one? No, right? We've done it a lot of times. You know how a FASTQ file looks like. Okay. Because you know how to run FastQC. Random for, pick at least one sample or random for one sample. And if you don't remember how to run the command, you can figure it out. You have ways to figure it out. Is the text readable in the back? Should I go up a little bit? Problems running FastQC? I won't be here next week, so don't expect me to tell you how to do everything. We say that, right? Teach how to fish. Don't give the fish directly.
Here we can give the name, the above name, father, mother. So we can reuse that file.
Do you want us to save them
You need to provide what?
The file name.
No. Just run it. It would automatically produce the output where the FASTQ file is. That's fine.
So like in the above they have mentioned father file. So we can give the name.
You need to provide, to run FASTQ, you need to, FASTQC, you need to provide the FASTQ file.
Yes.
Yes.
So there is above in the another before this step there is one file.
There's no BAM files yet.
First you father.
When father add one file, someone specify father.
You can do whatever you, father, mother, proban. Just do at least one. I know you did the homework, so I don't need to tell you how to navigate folders. It's the last day. I assume you have 5 days to do homework. It's fine if you just do one. You done it? I'm so proud of you.
I just copied the PDF.
If one thing you're going to do right, at least at the end of this, is run FASTQ. At least. That's good.
No, we can just take the reference.
Father lowercase. Does it have autocomplete?
Father, mother, you have to give the name of the child, right? And surname is there.
Remember the trick with the tap. You can do autocomplete. You don't have to type everything. I'm assuming all of you were able to run FastQC, right? Good. So FastQC, you need to provide the path to the sample. And because all of you know did the homework, I don't need to explain you how folders, subfolders work. Bonnie is looking at me like, oh, you're too harsh. So if you run it, you're gonna. So what is the challenge that you had running this? Tell me. What has been the main challenge? Why you, if you got stuck, what was the reason that you got stuck and you weren't able to run it? Because this is one of the simplest tasks we're going to do today. What was the problem? I don't know, somebody had problems, right, running this, I'm assuming? No? So we provide the name of the program we are using, and the only argument that we're giving to the program is a FASTQ file. So we need to provide the location where that FASTQ file is, which is in the father folder, or mother program, whatever you choose, and a line, and then the name of that file. And that's what you see on the screen. And if you run it, you obtain the HTML report that we've seen 100 times. Anything that called your attention? So these samples, I forgot where they're coming from. Yeah, automatically generates a report in the folder where the files are.
Yes.
Anything you want to comment on? What do you think? They look good? They look bad?
They're good.
They're more or less good, yes. There's something weird though.
The GC content is
Yeah.
weird.
I don't know the reason. I need to tell you, by the way, this is whole exome sequencing, but we are not aligning the whole set. We are aligning only one chromosome. Data for one chromosome to simplify the process.
Yes. So the score is approximately 20. So this
You
is
mean
20.
40? You mean around 40?
So score is almost near about 30. So this is good enough for the.
So when you say score, you're giving only one number. Think about this. This is all the sequences are here, and there is it's a box plot or histogram on the verticals. So the average and median must be close to 40, right, or not?
Should be there in the range of 35,
30,
30.
Okay,
35.
it is good enough. I mean, sequence
So we can
is what we have. We don't have a choice today to
in
resequence or anything like that.
in the transcriptomics we usually do the trimming for these
Yes,
lower
but we
species.
are
So
not
here
going to
we
do
are
it.
not doing.
No.
So there is no adapter
Is
content.
there a
So
percentage
it's
of remaining sequences if deduplication is removed?
We are not going to remove them. We're going to just mark them, but it's the same thing at the end. So your question is if there's going to be enough left?
No, how many should be left? Like 70, 80%?
Oh, how much is the percentage of PCR duplication? I don't know in this case, but it's a good point. You always need to check that, right? So if your PCR duplication is too high, you may have a problem. It may be that the sample is very amounts, or you need to change something in the protocol, or there's a problem with the sample. So PCR duplication, it's important to check, definitely. The levels we're talking here, the percentage.
Like if there are not many duplicates, it's fine.
Yeah,
95%.
I mean, sometimes you have to do what you have to do, right? So questions? Should we move on or not? I don't know. I have a feeling nobody's telling me anything. I don't get feedback. Need feedback.
so can you explain this?
Yes,
Per tile sequence quality.
sure. Um, I'm getting a question to review some of these plots. Any questions about the first one? No, we've seen it before. It's telling us about the quality of all sequences per base. We generally want it on the green area. That's what FastQC is telling us. It doesn't need to be there necessarily, but and you know that Illumina should go as high as 40. I'm not sure why we see 42 here. Again, I obtained this data from somewhere, so I don't know how it was sequenced. The question was per tile sequence quality. This is the location where the sequences are coming from. If, for whatever reason, so coming from in the flow cell, if for whatever reason some area is bright, like concentrated bad sequencing in an area, it's going to be flag, telling you, oh, there's a problematic section in the flow cell, because we know the location. Yes. So it should be, ideally it should be just blue, right? There's nothing here to look at it. There's always something, but. So it seems that, I guess, the problem is around here. That area of the flow cell produce a bunch of bad reads or low quality reads. So next step.
So in every
Yes.
case, if you have adapter contamination, one or
Yes.
two adapters, then also we should skip the trimming part or
So a good point too here. I mentioned not to trim by quality, but why we don't trim by adapter contamination. Again, look at JTK documentation. They offer tools for some trimming. We don't. We assume that the BWA is going to soft clip the adapter. But it's true that the first basis of the adapter could also align to the genome and produce a call or something, right? Yeah, we don't trim.
like we should not use strict parameters at least for informatic, even if there is.
Sorry, what is the question?
We should not use stringent parameters.
No, I mean, again, check. I would recommend that I think JTK, they have other tools, or I remember reading about it.
Because we used to do trimming always.
Yeah, yeah, yeah. And for JTK, I wouldn't recommend it for that because you're modifying the read. You're not modifying the quality scores per se, but you're modifying the read. And it's good to have all the information that you have for all sides in that region. And I'm not going to call an expert either. So, I mean, what I do, I work in so many different things that I'm not going to call an expert of anything at the end of the day. I don't know what I am an expert of.
Then we need to align.
So we did a quick fast QC. In any case, because we are not going to trim and we don't have a good reason to believe they're terrible, we're going to have to work with these samples. We don't have anything else. We need to report our case to the clinician and tell him, oh, this is the disease-causing gene. So there's not much we can do now. So next step is going to be alignment. There's going to be a lot of text to read if you want to, and I recommend you do. For alignment, we're going to use BWA MEM, and we're going to use pipes again. The pipe symbol, the vertical character. So remember, we take the output of one thing and we give it to the next software to do something with it, and we give it to the next software with it. And we have three pieces, right? We have 1, 2, and the third one here at the end. Probably the ugliest and the one that you think, why we're doing all this, is this one here. This is needed for GATK. And I tell you all the things, what it means in the text, so I recommend you read it. But the main idea is you need to label the sample that it is, what platform produced it, and so on. You need to provide some information for GATK because it uses this.
In the text, they have mentioned the add2 uses 2 threads for sorting.
Yes, I ended up modifying that for alignment. Yes.
2 threads for sorting means?
Did I set sorting?
No.
Oh, I did. I didn't modify this.
Yes.
Let's see what happens. We run it. Initially, I decided that we would use thread is a way to parallelize the analysis. Thread meaning, imagine you have 2 CPUs. You can use both of them. There's software. Not all software can be parallelized. In this case, we know they can be both of them. So you can indicate how many CPUs you want to use. That's what we're doing. Both SAM-tools and BWA can be parallelized. If you have really enough compute power and you want to parallelize at big levels, you can indicate as many CPUs as you want, and it's going to go faster. That's the main idea. Dividing the job makes the thing go faster. Is that the question? I'm answering your question?
So here we are using 2 types of threads.
No. The thread means you are dividing the. I'm going to simplify it in a way that you are dividing the jobs into 2 sub-jobs to run them in parallel. That's a thread mean.
So we have run these files, unaligned files. Now this SAM sort will give you the output file. The output is mentioned as a U, right? Output
Read it.
unexpressed
You. So another important thing, if you have to repeat this in your computer, read the arguments, because the ones I'm using here is for simplicity sometimes and for the setting that we're working with. If you have more compute power, you want to parallelize even more if you can, right? So do it. You can probably work with compressed BAMs if you want to. There's a few things that you should test yourself and see what it works for your setting. What I'm indicating here is what we can call maybe the best practices steps for analysis, but the arguments, you need to adapt them to your situation. Like memory, I think this should be fine in any setting, but you can see if you get an improvement if you change it. So what we're doing here, any questions about any of the parameters are answered up there? But so all the arguments should be explained. Didn't I explain that one? Yeah, I did. Oh, sorry, I keep doing. So here. So, well, again, so. So, oh, again. Soft clipping is when the aligner is not able to align it, and then we are saying you need to consider that in the other aligner, in the other read. The complementary would be the other. So we have two reads. They map. You're soft clipping something in one, you need to consider soft clipping in the other one too.
Is there a reason we're sorting it out?
Yes. Let me run it. Is everybody able to run it?
Yeah, but it's taking a
Just
while.
one question here with combined, like earlier you've explained that like as a single.
Why we do it in a single command and not separate? Is that the question?
Why
And
do you
I'll answer
define
yours
it
too.
like HUS as combined, or it is like supposed to be put together, or like as a separate?
You could run it separately if you want to, but it actually is a little bit more complicated if you run every step separately here. So we're doing three things. First one, alignment, and it returns a sum. Nobody likes to work with sum because it's too big. You could transform it to a BAM file and compress it. That would be this, or not compress it in this case, but you could. And then we need to sort it. The question is why we sort. For quick access. And a lot of softwares that we're going to use downstream, they want the BAM file to be sorted and indexed. Did it finish for anybody? It should take 5 minutes, I think. Still running? And how long did it say? Because if you want to know how long it takes. Oh, it doesn't give you something readable? I thought it gives you something readable.
Wait, okay. 586 seconds. 508.
508. That's not 10 minutes, it's less, right? So it's 8 something. 9. It's going to take 9 minutes, more or less. Yep.
So we can do these multiple steps.
Separate.
We can also separately do this.
Yes. But I don't think it's more work to do them separately, if I tell you. Because you're writing intermediate files and you're going to need some view. At least you're going to combine two of them, I would say. But you can do all three together. I think it's more work if you try to do them separately. I know you prefer me to explain things. Sometimes I'm going to take a rest from now on, or every now and then, and I'll let you read. even if it's running, you can prepare the code for the exercise that you just have below. Oh, you're running all three? Then you can do. Oh, it's done? Yeah, then keep working with the next exercise. So we only aligned the first one. We need to align mother and proline. So go ahead and align them. Even if it's running, if your code is running, you can start typing the align process for the other two, or copy and pasting, right? Don't lie to me.
I tried to copy it, it's all gone. So do I have to type it again? I mean, like I was trying to copy,
Yes.
and the whole the whole command is gone.
Oh, you can do Control Z to go backwards, and it should come back. So if you go to the cell where by mistake you probably remove it in Control Z.
Command Z, I think.
Yeah, Command Z.
Or those who are
You need to be in the cell.
और एक मतलब 4-5 बस जब तक वो आता है तब तक कर लो और 10 बार कर लो पूरा.
Okay, I'll put it back on the screen so you can see it. Um, I'm surprised that the Control C doesn't work, because if you type something,
It's not working continue.
if I type something, I can do Control Z and remove it. I'd send you an email with the code.
I sent it.
Okay.
Already.
So it seems that it takes for some people in between 9 and 10 minutes. So again, work the code for the next step, because it's going to take a long time. That's what we could do. Probably we could finish this section, let it run, and then we go for lunch.
You have mentioned doing for other samples. Do we need to open a separate notebook and do it, or we cannot do it in the same file? We can.
About the other folder.
Yeah, the other folder.
I mean, you could work in the same document. It's not going to look
Create some
clean.
confusion. Yeah.
So you could copy the document and put it in the other.
Okay.
The other thing you need, the other folder only contains one sample. So you have to remove everything related to multiple samples.
Yes. Yes.
If we have time, we can work in the other document too.
Next. Next cell बना. उसी फोल्डर में जाएगा ना, मैप उसी फोल्डर में जाएगा. तुम्हारा जब फादर का फोल्डर में फादर जाएगा, मदर के फोल्डर में उसकी मैप वहीं जाएगी. उसके पास, क्योंकि तुम्हारे वही है ना. ऐसे ही एक होगा, दूसरा चला गया.
run every command which you mentioning, because like I was wondering, like if we have a terminal open separately, like if we have a terminal open separately,
Yeah.
can't we just like run on like one of the files while the Jupyter notebook is running this one?
Yeah, you could.
Yeah, okay, let's do that.
Yeah, but the problem is, don't do it, because there's going to be 40 people trying to run something. Let's do sequential.
Yeah,
I don't want to get that situation we got the other day that we were using 100% of everything, and it seems that it went even slower. I mean, we can. I don't know how much we're using now, but I'm sure we must be 100% too.
So who's been using code?
Oh no. Well, yeah. I don't know. It seems. And we don't see all CPUs. I don't remember how much. No, yeah, we see all the CPU here. All 48. All are busy. You can guess already how to align the other two, right? Be sure to modify this line. And also remember, you can press tab to autocomplete if you don't remember how the files are called. Oh no, no, that's true. No, we cannot because it's quotes. We cannot use tab for autocomplete because it's between quotes. So you need to double check that file names are correct. Yeah, file names are in lowercase, just in case anyone has doubts.
First letter, first capital and second small, lowercase.
Yep. Folder names uppercase, file names lowercase. Yeah.
So rest is
There's no reason for that, but
so rest is small.
yeah, here it doesn't matter if it's uppercase, lowercase. It doesn't make a difference. They need to be different values though compared with the other samples. You could have named them however you wanted. I see a lot of people in this room. Just to clarify one thing, look what I'm doing here. So the father is done, we know, but because there's a number next to it, is the fourth step that I run in this pipeline. Mother, that you have the asterisk, it means it's running. I'm good. But I'm also running the proband. It's not running, actually. It will run when this is done. So that's what I'm trying to you to do. So don't wait for this one to finish to code this and run it. Run them. They will run by order. When this is done, automatically we'll come here and run it. So when you have all three, you can have the break. So I didn't want you two guys to wait for this to finish. I'm taking the break. I'm not I have the feeling that you guys are self-sufficient, right? Are you moving along with the notebook? Oh, that's good. So all the alignments finish well? Okay, good. So next steps, marking duplicates. So we are not removing them, we're just marking them in the alignment that they're saying this is a duplicate. So later on, they are not considered in the analysis. So I'm going to change the strategy. I'm going to walk around and help if I can. If not, you can keep going with the notebook, at least for this step, marking duplicates. You can run the first one and then run for, and we follow the same pattern, right? It's pretty easy. When you run once, just duplicate the code and run for the others. But please read, if you are curious about the options, read them. I generally put a table on top. And I don't know if you remember the first day, you were asking how we make tables, right? If you double click that cell, you're going to see how complicated those tables can be typing in Markdown. You know how I did it? I asked ChatGPT, put this on a table. And it's interesting how ChatGPT learns too. When you put something on a table, you keep asking questions, you ask for another table, it gives you back already in the format to paste it directly here. Oh, I had an error. Oh, of course. You guys are working with marked duplicates? Everybody finish marking duplicates. Can you raise the hand? Or the other way around, raise the hand if you haven't yet. Oh, you still not? Okay, keep going. So there's only one. So if everybody, or almost everybody, has run...
She has done.
Oh, you're done? Okay. So before we move on, did you run this command?
Yes.
Okay. So do you know what the second line does? Or the third line?
It's
Nothing. It does nothing. It does nothing because it has the symbol. Okay. Originally, I thought it would be a good idea to provide more reports, but some of these things were failing in the way that we install packages. So I said, okay, I'm not going to do it. But if you want a quick report of how the alignment went, you can do this flag stat, which basically reads the BAM file and gives you a report. So let's run it, and we're going to open this file.
yeah, father did he deduplication only again. He didn't read only. And it's indexed file. BAI.
Yeah, the text, the text that you see just above it is assuming that we run everything here, which we are not going to do it. I mean, we can give it a try, but the plots were giving me so much trouble that I think so I have to command the line, and I couldn't run it. So it may work in your system, so I'll leave them there just in case you need them. But be aware that if you run it, it's not going to produce the plots that mentions the text. So if we open the father, yes, because the extension currently is flagstat. If you it opens. I thought it wouldn't open. Yeah, it opens. I think we saw a similar report when we did the transcriptomics, when we aligned with HiSat2. But you can get a report of how many map, etc. And because we run this, does it tell you duplicates? Yes.
It's on
No.
zero. Everything is on zero in second. After plus. After plus, everything is zero.
I forgot what the plus thing means. I'll check it. But you get, because we're running after the duplicates, marking duplicates, you'll see them here. If you run this before you mark duplicates, they should be 0, 0. You get really good mapping. They're properly paired. So everything looks pretty much okay. I mean, the question would be if this is too much PCR duplicates. But I mean, we don't have a choice. That's our sample, so we're going to have to deal with it. Oh, no, it tells you what it is. So the plus sign is the ones that fail the quality. Any questions about this file? That would be a quick way to get a report of the alignment process. Well, at some point you want to know if you got a good mapping, right? If there's any problems in your samples or not. In the past I mentioned this before, like we process samples. If we see a problem, we go back and try to answer the question instead of, we don't run FastQC. If I tell you the truth, we don't run it. We only run it when we see a problem. So every time you see a problem, you need to go investigation, right? You need to figure out why. And if we get bad mapping quality, if we stop here and say, oh, the mapping quality, the percentage is really terrible, we go back to the FastQC and try to figure out maybe the sequencing quality was terrible. In any case, nowadays, every company that you sequence from, they're going to do some pre-filtering, or they're going to check the quality of your sequences. The mark duplicates also give you a report. I'm assuming it should be similar to this, which is 2 million. Yes. Does it? It's hard to read. This is a table, right? So, what are the duplicates? Oh, it gives you optical duplicates too. So actually, the software can give you optical duplicates. Or is the 0? Yeah, it's really hard without converting this into a table. 27% percentage duplicates, but we already saw that here, right? We have that number of duplicates, which more or less is 25%, right, of the total. Now, the next section is just saving a space. What we're doing is removing a file that we're not going to use anymore, which is the file before PCR removal, PCR mark duplication. Step 3, you started the step 3 already? Step 3 is 2 sub-steps. There are 2 processes we need to do. And the problem we had here it was that you ran the second one before the first one. And again, you're going to have to do the same thing for the mother and the problem. And each of those has 2 steps too. Don't forget the second one.
Everywhere in starting we have this. I've gone to this fourth one.
Probably mentioned it, but I'll mention it again. One thing that you didn't have to do for these exercises was to download the genome, create the indices for the genome. I already did that for you guys, and it's in one folder. Because if everybody does the same thing, if you look at the size of those genomes, are big. first, I got a few questions about how the code gets executed. So when you execute a cell, each line gets executed one after another. And if you go to a terminal, that's going to happen too. If you put all the code in a text file and execute the file, it gets executed from top to bottom. That's the way that it gets executed. The cells, you could execute the bottom one first and then the up. So you can choose how you execute things. The way the code is built here is to execute from top to bottom, the cells. Because some files get removed, some files are created, so that's what it makes more sense. Within a cell is the same thing. The code gets line by line, gets executed. We could have built, all the code that you see here could have built in a single cell. It would be hard to read, but it could have built in that way. I'm going to explain the third step just to review it again, because I know this is new and just looking at the slides and then writing the code are two different things. What we're doing here, we are not touching the alignment. So we come from the step of marking duplicates. So in marking duplicates, we just indicate it in the alignment if that sequence is a duplicate or not. That's the only thing we do. We have an alignment that now we're going to use for recalibrating quality scores of the bases. So we assume that the Illumina machine that gives us the FASTQ and assign quality scores is not doing a good job. That's what we're saying. So let's recalibrate them, because that's going to be important when we do the variant calling. I explained that in the slides. I was giving an example. When you see a difference, and that difference exists in a database because it's in the population, for example, right? We're going to assume that that's probably real. If it exists in another person and we see it in whatever we're sequencing, probably it's real, and we want to say, this is fine, don't touch it. If we see a difference and it's not present in any database, we're going to do a recalibration if necessary. I'm not saying it happens, but we can do a recalibration. The recalibration consists in different things. It's going to check if the quality on that area of the read is good or bad, right? Not only of that base, it's going to check the neighboring sites. Is a good region that the other reads also support the change or not? Is the quality of the reads good or not? It takes into account several things, and it will adjust the quality scores that was given by the sequencer. That's the only thing we're doing here. So in this code here, the base recalibration, which is the first step of base recalibration, because there are two of them. One of them is creating the model, and the other one is applying your findings. In here, all these lines is the databases we are checking for the presence of known sites, the ones that are in populations. This, we mentioned it before, dbSNP is a really popular one, is where there are a lot of annotations. This was a project where they sequenced 1,000 people. This is another database. I don't remember what includes this. This other line here, I'm limiting. I'm probably explaining this here, right? I don't think I'm saying anything new. You just need to read it. But just in case it's not clear enough in the text, this line, we are limiting the whole process only to the coding targets. I don't care if it's intron. I don't care if it's intergenic. Please just do the job in that area. I don't care about the rest. Why I'm saying that, if I'm doing whole exome sequencing, even if when you do whole exome sequencing, it's generally a process of capturing reads. The reads, they don't finish exactly, capturing DNA fragments. The DNA fragments, when you fragment it, they don't finish exactly what the exon finishes. You capture a fragment. That fragment can be part of intronic, part exonic. Then when you sequence a fragment, you're going to get the exon and part of the intron. Even when we do this, we see mitochondrial genes present here in high amounts. So what I'm asking is ignore the intronic and just focus on the exons. We actually don't ignore completely the intronic region. We take a few bases off. So even it says coding targets, it's coding plus something else. No. In these sites, they don't recalibrate anything. In these sites, it's just a database of known variants that we're going to assume they are real. If we see them in our sample, we're going to also assume that it's real because it exists in the database. That's the only thing. So we're going to say, it is in my sample, don't touch it. It's a correct SNP, so we're not going to recalibrate anything on that. But if it's not present in the database, let's be sure that the quality score of that variant is correct. And if we can touch it or manipulate it or adjust it, we're going to adjust it. So the second part, the only thing we're doing is whatever we've done in the previous one, we're going to apply it to our alignment. So now we went from the deduplication alignment to a recalibrated alignment. And recalibrated, I mean the basis quality scores, nothing else. The alignment is untouched. The alignment is done, and we're not going to touch it. So the other concept that I explained about readjusting the alignment is not going to happen. It was just an example to show one of the problems we had to deal in the past that the JTK doesn't do anymore. I mean, some tools index is pretty obvious. It's the same concept about indexing our genome. We want to create an index of a book. Our book is the alignment, so we can quick access the chromosomes. So if you ask, go to chromosome 7 position 1000, if you have an index, it would go faster to that position, whatever we need to do in that position. So you need to repeat the 2 previous steps for the mother and the proband. For now, we are working every file independently.
And after that, there is another formula where we have static quantized.
Read it. It's probably explained. It's not?
So it's we have marked duplicates. We have recalibrated our base codes.
Yeah.
So now what we are doing here is using this calibrated file.
Yeah.
And then we are dividing into some bins. So what we are doing in this 9 code, here what we are doing.
The last line of this 17 number on your
Okay, so it's not clear enough what I'm saying here. That's what you're saying.
Yes.
Okay. So the quality scores, instead of being precise, this quality score is 39. We're saying it belongs to this bin. So it's 30 something. And we don't care if it's 39 or 30 or 40 or whatever.
So we are dividing into discrete
Bins. Yeah, so we don't care if it's exactly 39. It belongs to that bin. It's fine enough.
So this is for the size reduction.
I know those parameters are a bit technical. I always recommend to go to the manual to get more details. Come on. And we've seen a bunch of times. RM stands for removing. We're just cleaning space. Oh, and again, I'm repeating myself. Is there?
That's what you were telling. Does it the sequence or the line? Using the fidelity of your database structures. जो place आपने call की हाँ
Let me know. Good.
ये बहुत second
which is supposed to be the annotated table, right? This is hard to read. We're not going to use that. That table is also missing the genotypes. And we produce also the genotypes, right? I don't remember how we call them. Did we? How we call? Final filter, right? Final genotypes. Oh, that one. Yes, yeah. So here we have the genotypes for father, mother, and proband, right? Which are not in our table, annoyingly, and we need them. But we reach this point. Now, the question is, I give you here the information about the trio. The proband, which is the affected, has this disease, right? The fragile bones disease. We know the father and the mother are not affected, so one of the exercises we need to do is to figure out what is the most likely mode of inheritance here. Any takers? You can answer. Go ahead. Homozygous, autosomal recessive could be an option, right? Assuming, so getting one allele from mother and father.
So neither of them is having a disease.
Neither of them is having, so that seems reasonable. We have more information. They are consanguineous. So that it means that most likely they're sharing the exact same variant. They're giving to the kid the exact same variant. But because it could be a de novo too, right?
Yeah.
Or I don't know if so that could exist, but we know that because they are consanguineous, they probably give to the kid the exact same variant because they share it. So now, how we filter? For the people that is completely done with this, there's another notebook which is called Using R to Filter the Variant, and it's going to read the full table plus the genotypes, and it's going to merge them. So you will get a table that is much easier to read. So there's a section here about plotting and filtering. That is just a practice. It is not going to tell you what is the disease-causing variant. This is just to practice. After that, you need to filter yourself that table to figure out what is the disease-causing variant.
Yeah, one question about my BPA results. So, like, is it normal that I don't have anything in the consequences section? Like,
Yes, it could be.
completely blank?
It could be. Or it's very long. Yeah, don't worry about it. You can have a really long answer, or empty, or yeah, it could be. If it's an intronic variant, you're not going to get anything there.
Yeah, it's like everything, like pretty much all the sections are empty except for the results.
Don't worry, you're not going to use the consequence in this case.
Yeah.
So I guess my point here probably, if you need to start with an annotation tool, I start with ANNOVAR, not VEP. Oh, another thing. You can go to the VEP website and upload your BCF, and it would be also annotated. You could do that too.
Why does it say there is no package called as AYPLR? There is no package.
Oh. Sorry, what?
I don't know what I'm going to do here.
You already done it. So this is annoying. I think a lot of people went to the R section now, right? I know it's hard to, especially at the beginning. The only thing we're doing at the beginning is to merge two tables in one, because BEP, when annotates and creates a table, annoyingly doesn't include the genotypes. And there is a typo in the notebook. Here when it says GT, what I meant is geno. That to check that we correctly loaded the genotypes for mother, father, and proband. And then I merged both tables. I checked that both tables are correct. So now we have a table with all the annotation, all the columns with annotations. At the very end,
It's not visible there.
oh, why nobody tells me about it? I'm here just talking. Should we solve the case together?
Yes, sir.
So did everybody or most of the people reach at this point where we have a merge data frame with all the annotation, only including the canonical transcript? Yep. So any suggestions on how to filter the table? Except that section there. I don't want to start with that one. So I'm at the point where I have this table. Right? We have our variants that are annotated, and we have the genotypes of each at the very end. Okay, we have all the column names. You have them, the column names, you have them a little bit up the notebook. Now I'm asking, we have that trio, and we need to find the disease-causing variant. Anyone can give me a suggestion on how I'm going to be filtering this table?
We have to filter according to the clinical significance or according
That's an option, but you have a table annotated. Can you be specific about what column you're going to take in that table that we can use for filtering? We know what we think about, what we know about this. We are assuming impact could be an option.
You probably want to first filter out by consequences, right? Like whichever consequence you want to take a look at.
I wouldn't do that. We can do it now. Okay, let's just start with that. So everybody comes out with that. It sounds good to me. But I don't know if I would start with that. We are assuming, you're assuming, we are going for the long-hanging fruit if we do that, right? If we are lucky, that's going to work. But sounds good. Okay, let's just start with that. So what we said, we're going to filter. We've been filtering every single day in R that we use R, right? We use that filter function. We need to give him a column name. How is called the column name that we're filtering? What's the name of that column?
name of the column.
Yes. Impact. Yeah. It's called impact. And what is the value that we want to keep?
Impact.
It's in the notebook. Don't look at me because I don't even know. You want me to filter for impact, so which values should I keep in the table? No, I wouldn't. We wanted to throw away modify. Oh, you want to filter as okay, little by little. You don't want to go directly to the highest consequence.
High.
You want to keep two?
High and moderate. But the high will be better. High impact.
Should we go for high? We can go for high. Okay. So they decided to go for high. That sounds good. And see if we are lucky, right? So now we started with 6,264. Let's check how many we have left. 18. Wow. Okay. We could probably manually check one by one, and maybe we find it if it's in here. But what else we know about this?
Then the clinical significance, whether among these 18 genes.
But we don't go even there yet. No. But what is do you have a column here called clinical significance? Do you?
Where in the column? Yes, so
What is the? I don't know. I'm asking for a column name here that we can use.
It has
Okay.
the
Sure.
clinical significance. So this is clean underscore significance.
Sure.
So it could be a pathogenic, non-pathogenic.
So you're assuming that the variant we're looking at is present in clean bar. Is it?
No, but first.
Well, sure. No, let's do it. Sounds good to me. Let's do it. So clean is significant. What are we looking at? So you're telling me that I should. So merge canon, right? I'm filtering again. Oh, this is not going to work because I'm telling you it's not going to work. So I'm not going to. Oh, I don't know why I did this, but I'm not going to erase the. So filter clinical significance equal to what? What do you want me to filter for?
Likely pathogenic.
Just likely pathogenic? Okay. It's called sorry.
Likely.
Let me copy and paste it in case I do typos. Did I miss it? So I already tell you why it's not going to work. Oh, we don't have it. No, there is one, but I don't know how it's called. So I'm going to have to. I forgot how it's spelled. But can you guess why it's not going to work? What you're asking for? Can you have an idea?
Based on this ID. This
No.
reference data.
So you're asking, yes. You're pushing way too much clinical bar. I'm going to tell you, it's not present in clean bar, that variant. It's not. So it's not going to work. I mean, you could do it, and if you're lucky and it's present, sure. But likely pathogenic is not going to work either, because you have pathogenics too. Yeah, but it's not annotated. I'm telling you now that it's not annotated. You have something simpler for filtering all this. We know that it needs to be homozygous in the proband and heterozygous in the father. Let's filter for that, right? So what we know that I mean we can even now. Right. I think it's used this. It's one or two. I always forget what R uses. So we know this. Father needs to be heterozygous. Or at least this is most likely explanation that we're finding, right? Mother needs to be heterozygous. Sorry, not comma. And the proband, how it needs to be. What I need to place here.
Recessive, if it is pathogenic, then the proband would be recessive.
Yeah, that's what we decided, that it would be autosomal recessive.
Homozygous.
Yes. What should I?
One by one.
Yep, correct. Sorry, I don't remember if in R is double and or it's not going to work. Sorry. It's a single one. I always have a No, either. Okay. Can you somebody remind me how we do N?
Proband spelling is wrong, and do we put and?
Oh, is that what it is? What I'm doing? Sorry.
No. Pro, P-R-O. P, P-R. Yes. नहीं आ गया
Okay.
It's coming.
We have two variants that, based on our filtering, has a high impact. They follow the expected genotype. We should check also what else. We know that it needs to be a rare variant, right? So all these are frequencies of variants in the population. It seems that the second one, it's too high to be responsible for this, right? 28% of the African, I think this is African, 24 American Africans is 45. This is not responsible. The second one is too frequent in the population to be the disease-causing variant. Could it be the first one?
Like four, I think. I scrolled down and like there is.
Oh, there are four?
Yeah, I scrolled down and like.
Oh, yeah, because of the concept. Okay. Oh, yeah. That's annoying. You're right. So which one is causing the problem? Is domains, right? So let me split by line. Select. If you do this, that's a trick. I'm removing the column domains, which is annoying. Oh. Wasn't called domains?
Domains.
Yeah.
In caps.
I know, I copy pasted, so I thought this would. Oh, now it worked. Okay. So we have four. High impact. What else we know? Oh, they follow the pattern of inheritance, the mode of inheritance that we wanted. We know that it's a rare disease. So we can still filtering in R, although four variants. I mean, we could manually check which of those could be the most likely, but let's keep filtering. All these are frequencies. Remember we talked about nomad? These are frequencies in populations that in theory are healthy, or at least represents a population. I don't know if they have a global one in VAEP. Yeah, okay, we have the max. We probably should filter for this, right? Oh, that's max in Africans, or is? Forgot what AAF means. No, this is allele frequency. I think we could filter for this. I'm assuming. Oh, but there is one that doesn't show up in here. Okay, I'm going to take the max one. Okay, let's add another filter. What frequency we should use? Any suggestions? Our variant.
We can filter.
Yes, that's what I'm saying. Definitely we can filter. Nothing is going to stop us. Any suggestions about 0.01? That 10%?
0.0.
1%?
Yes.
Sure. Sounds good to me. So smaller than 0.01. Okay, I did something. Oh, that's bigger. Huh. All of them are bigger. No, there's something. I'm doing something wrong, right? I'm choosing the right. I'm messing up if I'm choosing the right column. Oh, it's taking as a character. It's not taking as a numerical. Okay, this is going to be an issue. Okay, let's do it manually because we only have 4, right? We don't have to use R for this. So can it be the first one? If we look at the maximum frequency in the population, if there's a population with 15% of this variant, you think it's going to be the disease causing? No. This one says 45. You think it's going to be disease causing? No. What about this one? This seems pretty low, right? So what about the fourth one? 25%? Unlikely, right? So it seems that the way we filter it, there's only one possible candidate. Okay, let's go back. So this is the variant. So the gene is called CA2. What do you think? Could it be responsible for this disease? No? But you sound really convincing.
Because osteopetrosis is caused by, if it is osteopetrosis, then it's
Yep.
the other.
Okay, we have it. How many people think this has chances of being candidate or not? CA2. You need to think as an investigator, not just jump. I mean, we can check. Should we? There's a database called OMIM. Let's look at it if it's present in OMIM and we have information about it. See, OMIM, we talk about this, is a database for rare diseases. It may have information about if it's associated or not. So this is the entrance for that gene. And it gives you possible phenotypes. Let's do osteopetrosis, autosomal recessive 3 with renal tubular acidosis. Definitely it's not associated with the disease. It is, right?
It is.
Yeah. And inheritance mode matches our expectations, autosomal recessive. Yes, but there's a good point of checking ClinVar, right? You could also check if it's present in ClinVar. Let's see if it has a DB-SNP. We have a DB-SNP. Let's go to ClinVar.
Yes, it's involved in the pathogenic mechanism, the carbonic anhydrase.
Okay.
So there's a defect in the osteoclast formation in osteopetrosis.
Our was G2A, I think, right? There's no entry in ClinVar. The SNP exists, but there's no entry. In ClinVar, if we filter for ClinVar, we would have removed this variant because there's no entry in ClinVar for it.
Not entry, right?
No. So it hasn't been reported as disease causing in ClinVar.
So, but the stop codon, but the frequency is quite low. So, whether we should go about the function according to the function when we want to select some gene, one gene.
Yep.
That we can go back and we can look into the pathogenesis of the disease. But if we want to select one gene which could be of clinical importance, so it's whether we should select based on
This is a gene of clinical importance. It's autosomal recessive, right? We know that.
Okay.
With a stop gain, this could be clearly of the clinical relevance for sure.
So, but the frequency was quite low. It
Which is
was
great.
10⁻⁵.
That's what we want. We want to be low frequency. That's what we want.
Okay, I got it.
Because if it was a high frequency, it would be.
The
it would be that everybody. Yeah. You always need to think about the prevalence of the disease. So if your disease is really rare, you could push even more that value. Of course, of course, you are always limited to the size of the database, right? If the database is big enough, you can reduce even the filtering to lower values. If the database, I mean, you can keep pushing that 0.01%, right? Even lower if the database allows you that resolution. But if you're filtering against 1,000 individuals, it's always hard to go lower than those frequencies we're using. But anyway, we found it.
Franklin's was the same as pathogenic strong. The CA2 gene mutation, it's pathogenic strong on Franklin.
Sorry, I cannot hear you.
It's showing its pathogenic strong on Franklin. The RS
Oh,
is
good.
getting showed on CleanBar now.
Yeah, I went to CleanBar, it's not present there. Oh, good.
So I went to Gene B. So Gene B like showed. So from there I got this NMID and everything. So
Okay.
it's pathogenic strong. That's what
So Franklin is better.
Don't know, but it's not showing any in silico predictions.
But nobody in Franklin. Can you get if anybody predicted or is just it's
See,
Franklin just saying.
Publications, yes. I think it's saying because of null variant and that's it. This is a 3 also. Osteoporosis, it's showing. That's what you were saying. It has Brazilian bone disease.
Yep. Okay. So what's the question? You mean?
No, I'm saying that on CleanBar it was not showing, but here it's showing.
It shows, yeah, but I mean, if it shows here.
Yeah, that's what I'm saying.
But I mean, it's likely pathogenic based on the classification, but there is not reporting, right?
Yeah.
Nobody
Then why
reports
it's
0
showing
cases,
that,
right?
Yes. Then why
So,
it's showing that it is linked to autosomal recessive osteoporosis?
oh, in where? Sorry, there's a.
Yes. In conditions
Oh,
associated with
because
CA2.
it's thinking about the gene.
The
Only.
gene.
It thinks
But
about the
this
gene.
mutant has not been reported.
Yeah, it hasn't been. So it's the same situation as in CleanBar. You also have another folder if you want to practice. If you want a different disease, you can do PKD. Is the other folder.
So finally we come to the end of this 5-day course, and we must all be tired and exhausted by now, both mentally and physically. So I do hope that you take something back from this workshop, because both Frank and Bonnie have made a lot of effort in organizing this, so that you derive, you get as much exposure to all kinds of genomics, both transcriptomics, single cell. So I hope it was useful for everyone, and you'll be able to use it for your research or your clinical practice. Anyone else, anyone from the participants wants to make any comment?
It was really commendable effort from both of you. You were just till the last moment. I was telling somebody, tell him to stop, but you were with the,
Oh, I'm here, right?
Yeah.
So it was great learning experience, because we were blank. We were not even considered as beginners, so we don't know anything. Earlier we were not aware of anything about the data analysis. Now we can try at least not to implement.
You have the courage to try at least now.
Yeah, pretty much. Thank you. Like I didn't have a lot of biological background, so I often had to Google stuff during the workshop. But I think I was able to follow thanks to you. And at least the resources you provided were really good.
Good. Thank you for putting up with me. And also, I'm happy that everybody came here with the homework done before they came. So that's really great. You're really, really nice, especially if I lost my patient at some point. Thank you for tolerating that too. It's been an experience for me, definitely. I learned a lot preparing the workshop, because it was a different situation that I'm normally working on. So this was great. Thank you very much.
I mean, when almost 2 years back, when Amitji and Manpreet came and asked me about this, I think it's 3 years almost, not 2 years maybe.
It was the month of November, 2022.
Yeah, it's almost like 3 years back. So one of the things that I thought was bioinformatics may be a good thing to do. And one reason that why I always thought just talks sometimes is not worth. I mean, it's good. People come and give talks. It's not bad idea. But then talks have less learning. I mean, you listen stories, and as a scientist, we scientists are bad at giving talks. The reason why we are bad at giving talks, because we tend to focus a lot on our individual work, like scientific work, and we actually go into nitty gritty that this protein does that, and this RNA does this kind of story. So end of the day, and if you are not from that field and you are not interested in that, then end up being just after some time you just shut yourself up, and then you are just wasting your time and everybody's time. So that is when we have to do hands-on. That might be the one good idea. And then I also thought about, why don't we have a hands-on on NGS-based methods like library prep, like ChIP-seq, ATAC-seq, RNA-seq, library prep, and sequencing. So that idea was actually a good idea, and I could have actually bought a couple of my folks from my group, like Irina or Chris or somebody, to help me out. But then the problem is finding reagents. You have to spend a lot of money to make that happen, because you cannot just show if you have a hands-on, then I want all the participants to make their library, sequence them, and at least look at sequence how it is looking like. And to happen that, you need a lot of money. So which was not a good idea. So then this whole idea of bioinformatics came. I actually, to be honest with you, I reluctantly asked Francis, because I was actually, we both were actually doing few bioinformatics workshop for YCGA, and Francis was always leading some of those efforts. So at some point I just asked him, like, would you be willing to? Because this is a lot of work. I mean, so there are two aspects of that work part. One is a lot of work in sense, like I know that he has to travel almost like 14 days, stay in India. He never came to India, so I was worried about food, environment, everything. And then because when you are teaching whole day, you have to be physically fit to do so. And then any kind of eventuality coming out of food or water will be much of a concern for me. So I was like, initially I was pretty reluctant to do so, but then I thought still I asked him, and he agreed to it. And then the amount of effort Francis had put into all these things, like almost last 1 month, he was just making all these Jupyter notebooks and the notes and everything. So a lot of effort went behind the scene from his side. And I was feeling bad about it. And this was a time, unfortunately, in my life, I was actually in middle of a lot of things, lot of personal and professional thing. I'm in middle of that. So I don't know how much I could have actually helped him in many of those things, but still I could not do anything about. And as you saw, like many of the presentation and worksheets that I was teaching, I essentially made here when I came. You know, and Francis was every time he was poking me, I want your Jupyter notebook, where is it? And then I was, I'm working on it, but it never happened, unfortunately, because as I said, like so many things were happening in my life. So, but I'm really happy for one thing, that the way this workshop went, um, in spite of all the issues that I thought about may happen, but nothing happened really. So it's really nice, and you all stayed till all this time. So now, the final one thing that I would like to say, what I think you should be taking away from this workshop. So many of you may be able to do coding. At least you will be able to modify the code given to you and could run your sample. But even if you could not do that, even if you could do that, that is the ultimate success of this workshop. Okay, even one of you could do that, that itself is a big success. But what else we actually need to know, what I actually want you to take from here, that next time when you see a data, you should understand that how this data was created, what was the assumption behind it, what you should not infer from this data, what is the actual interpretation should be, what should not be also. Like I don't want, some of you will be actually making a presentation and saying like, this gene is more significant because it has more lower p-value. That is the last thing I want to hear from any one of you, because it is significant or not significant. It has no more significant or less significant. So those kind of, because how much how much time was spent on just explaining DEC2. Now you know what goes behind those data sets. What are the quality parameters you should be looking for? What are the different ways that data can be analyzed and interpreted? So now, if you understand at least that, and then you understand that when you see a data, as well as you actually explain that to a bioinformatician, saying like, hey, I want this to be done. Can you do this way? Because, and then you can talk to those folks also who are doing bioinformatics. Otherwise, you will say, I want to do an RNA-seq analysis. What does that mean? Okay, so they will give you something back, and then you have, neither that person has an idea, neither you. So if you can talk to a bioinformatician now, from whatever you have learned from 5 days, you can make sense out of your data, then I think that itself is a success of this. And then above that, if some of you can even run the code that was given to you for your own data, that is the most amazing thing that can happen. So again, thank you so much for being an amazing student, and thank you Amitji and Manpreet being such a wonderful host. I really had a lot of fun. I happened to actually go with Manpreet and Manpreet's husband many times till late night we were out. So it was really fun. I think, yeah, it was, and definitely Jomin and then Francis is going to see a little more of India. I hope they will survive next 5 days. So Francis survived first 5 days. The last 5 days I have to see how he does. Yeah. No, anyway, thank you so much. Thank you for all this amazing workshop. I really enjoyed it being here, and I think you also had same experience. Okay. Thank you.
This kind of hands-on workshop was the first experience for you guys also.
No,
They're doing it in US.
US regularly, right?
Yeah.
Yeah, for me at least, the experience comes from all the setting, right? It's AWS. I used it in workshops like 16 years ago, but I haven't touched it anymore, right? Because in Yale we use whatever we have locally, right?
So
coming here, preparing everything, be sure that we connected in Mumbai and everything, all preparing the materials, preparing Jupyter labs and all that.
So even we thought that we will be using that Nabi has got a supercomputer. We tried that, but sadly it didn't work out. And then Frank suggested that we could use AWS. We were quite scared, but it would be very expensive. But then he told us that it might not work out to be that expensive.
Yeah, I mean, I think it worked great, right?
Yeah.
I mean, we only have maybe a couple of issues, a slowdown.
The first day, I think, when they were running bulks.
Yeah, I remember the day that we got stuck for a while, but not that bad.
So good thing about doing workshop in US, especially for EL when we do, that we have a lot of infrastructure support. We have a lot of infrastructure support. So like we have our own high performance computing system. So and a lot of people have already their accounts there. If not, then we have to just make some accounts, give access to them to that account, and then you'll also have something called as open on demand. So essentially it's a GUI thing where you enter and you just take the Jupyter notebook. So it's very easy to actually conduct a workshop. You don't have to do, so we have to do so many juggling here to get this happen, AWS and then putting those Jupyter notebook, make it work, and then speed, we don't know, with 40 people it will survive. And the other issue was the bandwidth of the internet here. So when 40 people are actually submitting their alignment, will the internet will take that much bandwidth or not? So there were those kind of,
It did.
yeah, it did actually. That, I mean, we did not see any because I was same time I was browsing and watching some news and everything, so it was working. That means, like, the internet was no net. So
It's the official. It's a government. It's a BSNL. So.
no, yeah, it is.
BSNL is not
So
that
those issues were real issues, and we have no answer to it unless we really test it in the real life, and we have no second chance, because whatever we decide, we come here and we see that BSNL is slowing down. We have no option other than saying, okay, half of you shut down and half of you work, and then after half an hour, other half will work. Something like where you innovate. So, but as I said, like, this was a experience we are uncertain, but what it turns out to be was excellent. Like, so everything, whatever we thought that may go wrong in this, did not happen that, and that is actually amazing thing about this. And I think I'm really happy, and I hope you all will be benefited from this, and then and be in touch. Okay, with me and Francis, you can find always internet somewhere. Our name will be there. So always reach out if you need any help, even for some small issues with code running or something like that. You have some questions, please. Yeah, you can ask me. I mean,
It's easy for him.
Are you blocking me?
No, it's exactly. It's easy for him. It's like he's going to change jobs and he's going to change email.
Yeah.
So you're not going to find him in a month from now.
no, I told you like, no, I told you like you can find me in internet. So yeah, I should just tell them that where I'm going. So
Exactly.
I'm actually, yeah, I'm going to actually move from Yale to a new place called as Varsity Blood Research Institute. It's a privately funded research institute, and I will have a joint appointment at Milwaukee Cancer Center and Wisconsin Medical Center. So any of those places you type, you will find my name. So see, I'm not going away. Okay. Yeah, no, it is indeed it's a great experience, and I hope you also had same. Okay. Thank you so much.
Can he wants to talk to?
Yes, you thank you. You all the way.
No, we don't have. You want to
Thanks for coming. Actually, I mean, yeah, even thanks for coming. Yeah, thank you. So we can now have the valedictory function where we'll give the certificates to the participants. So.
That's what everyone's waiting for. You all want
Oh, I see. I see. No, not because of us. We want to run like or from a moment to get the certificate there.
No, we won't motivate you.
I'm already.
We analyze data profile also. So for our
Oh, by the way, actually, I'm
Want to know that they can have the.
So actually, we have to thank two people. They, without them, this, who is the third one? Oh, okay, yeah, Vaishali also. Yeah. Yeah. Yeah, she was also. Yeah, no. Without them, you have seen like they were just walking around all the time. I think if they have a Fitbit or watch like that, she might have got like some 25,000, 30,000 steps on a single day. I mean, without them, this was not even possible. Yeah, they did a great job. Thank you so much. Thank you.
And to add to that, they are in the middle of submitting their PhD synopsis. So they've been really busy, and I told them you have to be here. So for those had no option.
Thanks for the effort,
So
definitely.
maybe you can just give them the certificates. Nirani, please do. No, no.
we should get a picture all together. Does anyone?